gentlemen, the books is in. The books are in. Yep. They in. They're here. What up? ETM Hotel, Brittany, Shawn. Welcome in peace. My name is Shawn. I'd like to thank everybody for tuning in to Science with Shawn. This is episode, 16? Man, I appreciate everybody that's been showing a little love and sharing the shows and trying to get the word out so that we can educate people and be accurate in our assessment regarding vaccine, the latest news, conspiracies, and the whole nine. With that being said, yesterday's show, I got some great feedback from some people, not even really my Facebook friends or anything like that. People that are just part of the actual, I guess, YouTube world that have been tuning in lately, trying to, I guess, get some information. And that's really what it's about. It's about sharing correct information and building with people on subject matters that are actually, about advancing the world. This is not a black problem or white problem. This is or Asian or, whatever. This is a whole world problem. We're in the middle of a pandemic, and, viable information is critical during these times. Correct information is critical during these times because people aren't just really accustomed to seeing these type of things transpire or used to using correct methods in order to come to an honest conclusion regarding vaccine sciences or just science in general. So, man, I like to just thank everybody for participating in it, liking the show, sharing the show. You know what I'm saying? Showing a little love. We're just trying to keep people thinking critically, right? We're supposed to be the conscious people, right? So we got to make them think critically, and we got to keep on using the correct method in order to come to an honest conclusion. We can't go off of conspiracies and hearsay. We got to live in the realm of evidence and facts, right? We got to collect all the data. You know what I'm saying? For and against. We don't have to be biased in our research. We can scrutinize anything. And if it stands tested or if it stands scrutiny, then, we accept it. If it don't stand scrutiny, we reject it. It's that simple. It's that simple. So again, let me see who we have in the building before we get started today. I just want to say peace, Brother Chavis. See you, Omar, Shaz, Pure Black, Nunu. I see you in the chat. Dave B, what up? You know what I'm saying? It's Monday. I know a lot of y'all might be on your holiday flow, and that's cool. You know what I'm saying? A lot of people might not even tune in. That's cool too. But if you do catch this replay, I appreciate it. Share it. Let's get this info out. Let's get it out. So real quick, before I just get started, let me share this link. And then we'll get into the bill. I know a lot of people working today, man, but y'all got earpods. Y'all got earpods. Put your earpod in, tune in, and just listen. Again, man, like the show, share the show. You know what I'm saying? I know a lot of people might still be asleep. We need to wake them up with this realness. Who else in there? What's good? What's going on, everybody? All right, so while we're trying to get people in the building, man, I'm going to go ahead and get this thing kicked off. I'm going to go ahead and share my screen. Let's talk about the latest news. All right, so Europe is beginning its vaccination rollout with a goal of inoculating 450 million against COVID-19. So while we dealing with conspiracy theories and fake deaths of nurses in Chattanooga and acting like, you know what I'm saying, they dead and all of that, and retractable needles, and people ain't really getting the vaccine or taking the vaccine is for show, or people falling out or having these rational allergic reactions. Europe is moving forward with a plan to roll out 450 million. You know what I'm saying? They are stepping up, and even though the published data is not out right now on AstraZeneca, um, they have already taken upon themselves to put themselves in line to go ahead and retrieve and purchase um, that vaccine as well. So they will have three vaccinations working in their favor among their people. Yep. So they like, look, we ain't playing no games. We getting rid of this thing. Straight up. Straight up. In the US, we only have 2 million people vaccinated, right? And the first people that were vaccinated have 2 more weeks before they have to receive their second boost. Some people. Some might be a little bit more ahead of the others, but uh if we're looking at the dates and all of that, you know what I'm saying? That's when they'll be receiving the vaccine in 2 weeks. However, let's go forward. All right, the European Union began a mass vaccination campaign on Sunday to eventually inoculate some 450 million people in 27 member states against COVID-19. EU leaders have negotiated contracts for more than 2 billion vaccine doses from various suppliers. The first 200 million doses are vaccines from Pfizer and BioNTech, which was a co-developed by a husband and wife team in Mainz, Germany. In Greece, the first recipient was Evstavia Camposoulou, an ICU nurse in Athens. Greece television stations broadcast the vaccination. Camposoulou, clad in the blue scrubs and a mask, gave a thumbs up, and a masked colleague sank the needle into her arm. Everyone clapped. All right, cool, we get that. Now, a nursing home resident followed. The Greek politicians, including Greek President Katerina Sakelopoulou, science is giving us the best gift this Christmas, she told reporters. Similar scenes were playing out across the EU, where more than 350,000 people have died of COVID-19. The hardest hit country has been Italy, which has recorded more than 70,000 corona deaths. 3 healthcare workers were the first to receive the vaccine there. Italy's foreign minister, Luigi Di Maio, yeah, that's how you say it, urged Italians to get vaccinated. Now, the COVID vaccine is the only weapon we have to stop the virus. Miano wrote on Facebook, slowly our freedom will return. All right. Oh, Lord, let me get this out here. Okay, cool. Now, that's what she's saying. Now, the European Medicine Agency based in the Netherlands greenlit the Pfizer-BioNTech vaccine December 21st. The first batch of shots was delivered Saturday, though the vaccine was delivered in Germany, which currently holds the EU's rotating presidency. The United States, United Kingdom, and Saudi Arabia all received it before the EU. The German magazine Der Spiegel accused the EU of dragging its feet and haggling over prices. EMA director Emir Cook told Euronews that the agency was doing its due diligence to shield taxpayers from liability and to make sure we have not missed anything. EU leaders wanted all 27 member states to start vaccinations on Sunday as a way to show unity. In a tweet on Saturday, EU Commission President Ursula von der Leyen called the synchronized rollout a European success story. but some countries couldn't wait. On Saturday, nursing home residents in Northeast Germany were immunized, including a 101-year-old Gertrude Haas. Hungary and Slovakia also started a day early. So it looks like Europe has already put itself in position to move far ahead than every other country or nation among themselves, right? Among its peers. This also goes to show the ignorance and intelligence of North Americans while we're caught up and wrapped up in craziness that actually can kill people by misinforming them. Now, I'm not going to read the whole entire article. You know how I roll. I get toward the end and then I let you get it. Of course, you know the sources all sources are in the description on the video. So moving on. South Korea to accelerate virus vaccine efforts as first UK variant detected. Remember earlier or last week, we talked about the actual new variant in the UK. Let's go ahead and add South Korea to the list. South Korean officials are vowing to speed up efforts to launch a public coronavirus vaccination program as the country on Monday announced it had detected its first case of the virus variant linked to the rapid rise in infections in Britain. The new variant, though, thought to be more transmittable than others currently circulating, was found in 3 people who had entered South Korea from London. On December 22nd, the Korean Disease Control and Prevention Agency said on Monday, South Korea will extend a ban on direct flights from Britain for another week until January 7th and will require any passengers arriving from the country or South Africa to undergo testing before departure, officials announced on Monday. Overall, the KDCA reported 808 cases of midnight Sunday. Now, these people were talking about something in Britain. Now they telling South Africans, you know what I'm saying? Because these people are from like the UK. All right, let's get that straight. They ain't really Africans. They're from the UK. By the way of colonization, they are in South Africa, and they are telling them, we know y'all go to UK, y'all go to Europe often, especially around these times. You need to make sure that you're safe before you come our way. So they banning travels from that area and South Africa. All right, that's peace. Now, overall, the KDC reported 808 new cases on Monday, midnight Sunday, the lowest since a record 1,241 infections were logged on Friday. Authorities cautioned that the drop may be due to the less testing done over the weekend and the Christmas holiday, and said on Sunday they would be extending social distance measures until early January. South Korea's government has faced mounting domestic criticism over its vaccine procurement and rollout plans, which call for the first vaccinations to begin in the first quarter next year, months after placing such as the United States and the European Union. All right, now, meanwhile, the first shipment of Moderna COVID-19 vaccine was distributed to 3 military treatment facilities across U.S. military bases in South Korea. U.S. Forces Korea said on Twitter they added that South Korea as one of 4 overseas locations in the Department of Defense initial phase distribution plan. Negative views of the vaccine plans was one of the leading reasons that drove President Moon Jae-in's disapproval rating on an all-time high of nearly 60%, posters Real Media said on Monday. So the people in his area, South Korea, they like, yo, nah, bro, we ain't rocking with you like that. That's how they said it. Now, regulators will shorten the period required to approve vaccines and treatments from an average of 180 days to as little as 40 days. The Minister of Food and Drug Safety announced on Sunday. An additional approval process to distribution and sale of vaccines, which usually takes several months, will be shortened to around 20 days, the minister said. Medical workers and early residents will begin receiving the vaccinations in February and plan to vaccinate the broader public are accelerating, Moon said on Monday. So he got his hands full. Like I said, I'm not going to read the entirety of it. It's down into the bottom. South Korea has issues. It has the new variant, which allows it to move quicker through its community. They are shutting off flights from the UK as well as South Africa to South Korea while they try to control it a different particular way. And then they have their vaccination rollout plans. Again, if you're just joining us, Please like the show, share the show. I appreciate everybody tuning in. Um, all of the sources that you'll see on my screen will be in the description of the video. Again, I see we got some new people up in the building. So what up, Zane? What up, Goddess Segment? What up, Corey? Concrete Corey. Gullah was good. Lamar was good. Let me get pure comment approved and Zane comment approved. Sorry about that, YouTube tripping. Yeah, well, I really don't even tune in to that, so I really don't know what happened over there yesterday, pure. Especially when it come to old boy that you just mentioned. Boss in the building was good. Was good. All right. So let's move on. Let's talk about us. All right. So the latest news with us goes that we have reached 330,000 deaths. Now, two articles ago, you heard me mention how many UK deaths there was, like over 350,000. We're at 330,000. We're right behind the UK with this thing. All right. So let's deal with that. The CDC on Sunday reported 18 million cases of new coronavirus, an increase of 179,000 cases from its previous count, and said deaths had risen by 1,309 to 330,901. The CDC reported its tallied a case of a respiratory illness known as COVID-19 as of 4 p.m. Eastern on Saturday versus its previous report a day earlier. The CDC figures do not necessarily reflect cases reported by individual states. All right. So that's just an update on where we are and where we're tracking right now with the COVID. We need to get on the game, man. We need to get on the ball and continue to do what we need to do to make sure that our people are abreast. Like I said, this article will be in the chat. And let's get into the meat and potatoes of the actual bill today. Before we do so, let me do this. What up, brother Morpheus in the building? It's good. It's good. Let me do this before we move on. Anybody got any questions regarding South Korea experiencing a new variant? Anything about the UK and its plans on rolling out, or anything that was mentioned in that article, or the one that I just read regarding the CDC? I'm looking in the chat, so sorry for the delay. All right, now see what's going on here before we get it cracking. Stop sharing my screen real quick. And yeah, man, like the show, share the show. Please share the show. You know what I'm saying? The more y'all like it and the more y'all do all of that, the better it is for all of us. So I don't see no questions regarding anything that was just read. So we kind of pretty much up on the latest news. Just to kind of expand on the news, we're waiting on AstraZeneca's data. Once we get the data, we'll be able to analyze it and we can, put it through the ringer. We can think more critically about the data. We can analyze it against its peers and do what needs to be done. All right. So how many variants are there now? Nah, it's the same variant. The latest is a new variant. But as far as just kind of, you know what I'm saying? It's the same virus. What we need to worry about, we don't need to worry about the variant. We need to worry about the strand. If a new strand, meaning that it mutated in a whole new strand and it has basically a new body type, that's what we need to worry about. Right now, this new variant is nothing but a different spark plugs. Yeah, AstraZeneca did. Yeah, I read that too. I read that article. Actually, in Reuters, they were talking about it. It was coming through as 100% against. But we need to continue to do all of that and deal with it. Hold on, just a second for me, family. Let me grab something. Even though I am not at work, I am really like at work. Craziness. These supervising folk, yo, they don't have what they need in order to be successful. So they call you. Even though you try to arm them before you take your little vacation. All right, so I'm checking it, Wolf. Checking it right now. See, before we get this thing cracking, hey man, listen, I'm telling y'all right now, we finna get it in. Oh yeah. I read that. I read that last week. So yeah, Morpheus referred to the LA Times article where the rich people are trying to pay $25,000 to skip the line to go ahead and get the vaccination so that they can continue to build and do the things that they need to build. However, while we're fighting conspiracy theories or dealing with conspiracy theories and the rejection of vaccinations, rich people, the wealthy people, are trying to get vaccinated so that they can go ahead and protect themselves. Whereas we got people that's saying that they don't even want to just get the vaccination. So there's really a huge a difference between us and the wealthy. The wealthy are like, we ain't playing these conspiracy games. We getting vaccinated. I'm gonna give you 25,000 to let me go ahead and get the shot. They want to skip the line. We, on the other hand, underprivileged, ain't even in line for the shot, ain't even nowhere near being told that the shot gonna be available to us, are rejecting it, creating conspiracies and coming up with excuses on why we shouldn't even have a conversation about vaccinations and or advance this information among our people. Like, we can't even have a scholastic conversation about educating our people on vaccinations because everybody want to water it down, pseudo it out, and then talk about, oh, well, this is how they treated us back in the day. So that's what's going on with that. David hit me up and said something a second. He said, did you build on Kalia Mitchell, the nurse in Tennessee? Said she got Bell's palsy. No, I haven't did that. You know what I'm saying? Your favorite rapper got Bell's palsy. Conway. He make a rap about it. He got Bell's palsy. You know what I'm saying? And he didn't get it from no vaccine. You know what I'm saying? He inherited after he was shot. You know, and he can't get rid of it. You know what I'm saying? But hold on 1 second and let me try to get with that. Because you really got to deal with this thing the way that it actually go. Facial nerve palsy. Circle individual. Nah, man, I don't even see. Hold up. But this is common in society. This ain't got nothing to do with the vaccine, though. We cannot say that. She just said she got Bell's palsy. She didn't say all the other stuff. Kalila. No, I ain't talk about her though. She a director of human resources. I don't know. Was she in Memphis? I'll get into that another time. I'd probably have to talk to Malcolm because it really ain't saying too much about her, Dave. I don't see any information regarding her talking about Bell's palsy. But right now it's common, you know what I'm saying? You got 2 million people that in America that have taken the vaccine. See the pill. Have tested positive COVID premiere. Wow, somebody else has tested positive for COVID, and it looked critical for them. Anyway, let me continue to read before we get going, man. Like the show, share the show. Nah, bro, I ain't tripping on that. Yeah, that's what it is. It's a nerve infection. What up, Mal? What's good, brother? Nah, I haven't seen it, so they probably made that up. Let me see. Um, nah, bro, I ain't get no video. This video right here, look, and I'm finna show you this right here. Hold up, I'm trying to check something. Let me stop right here. I know you ain't talking about this, yo. I know you ain't talking about this. You talking about this right here? Look at the writing. Did this nurse get injured by CV-19 shot? Bell's palsy side effect. Is this video real? It says allegedly. So when she takes the mask off, right? Let's see what it says.
The whole left side of my face, actually. I have Bell palsy now. And as you can see, I can't smile. I'm trying to smile. I'm just getting embarrassed.
Now, if she's a nurse, why we ain't talking about it? Like you said, she's a nurse from Tennessee, right? You see what I'm saying? Like, okay, so let's deal with it.
I am a registered nurse in Nashville, Tennessee.
All right. Now, you saying she from Nashville, Tennessee. Her name is Kalila Mitchell. Watch this. Heather on Twitter. This what they sending around. So there's the YouTube video. Oh, I ain't got Twitter, so they go to video again, right? She says
Hey, Nashville.
God, I hate when they do this, yo. See, it's the same video. The dude was sharing, right? It's still him. This is the same dude. All right. Facebook. Watch this. Now I got it on all news, right? So the news will report this if this is the case. She's supposed to be a nurse. Why is it a video of her out and not a news crew or anything like that? Now if she took this video and did it herself, then she gotta prove that she a nurse now. See? Everything is a hashtag, a video. You feel me? Everything is a hashtag, a video. This is the problem that I have. Like, yo, why? Let's get it all the way. If you're a nurse, right? Everything is a video. Let's deal with it. Like, can we deal with it? We can't even deal with it. Where is she a nurse at? So let's listen and let's see what she a nurse at.
And my name is Khalilah Mitchell. And my name is Khalilah Mitchell. I'm reaching out to everyone about the COVID-19 vaccination. I recently took the COVID-19 vaccination. After the shot, I felt fine, but within 3 days, I went to the doctor because I had problems with my face. The whole left side of my face, actually. I have Bell palsy now. And as you can see, I can't smile. I'm trying to smile. I'm just getting embarrassed. But I just want everyone to know that I think this vaccination is the worst thing ever. And I would not give this to anybody, even my worst enemy. Please, America, they do not care about us. Do not take this vaccination.
I'm sorry.
Nadal przywiązuje dużą wagę do swojego wyglądu.
All right, so we heard from the sister. Now, she does not state where she from. It does not state if I'm a nurse, it's not a side effect. It's not a side effect, Misha. Yeah, it'll wear off, but it's a nerve issue. Bell's palsy, let's look it up. Two million people have been vaccinated. 2 million people in America. The four people that had Bell's palsy or whatever, out of the trials, there was 70,000 people. Well, let's just say 44,000 people vaccinated in the Pfizer trials. 4 developed Bell's palsy. Well, 4 had Bell's palsy. All right, you had 3 from the vaccine group, 1 from the non-vax group. I'm gonna say it again: 3 from the vaccine group out of 44,000, and 1 from the non-vax group. Right? Okay. Moderna. Have we heard anything about Bell's palsy in Moderna? There was no significant signs of it causing Bell's palsy, but they're still doing all of that. Now, so what is Bell's palsy? Bell's palsy is a condition that causes a temporary weakness or paralysis of the muscle in the face. It can occur when the nerve that controls your facial muscles becomes inflamed, swollen, or compressed. The condition causes one side of your face to drop or become stiff. All right. Misha work in the hospital. She know that it you know what I'm saying, it'll be a little tenderness and then it'll come back or whatnot. Okay, whatever. So she got to prove to me that she a nurse. So the FDA's watching for it. They all on it. I don't know this lady. I don't know what hospital she work at or anything like that. And I would need more evidence of this lady being a nurse. Her name is not on the nursing list, not in Tennessee. Oh, it's over. Let's see. Let's see if we can bag Malcolm up.
Peace, Shawn. Peace to the chat.
Let's see if we can back him up. I don't know if we have access to the board of nursing or the licensure of nursing, but I want to show y'all what I'm doing with y'all. But I want to get into this. So the board of nursing, I don't even know if we can get into the members, but if you are, you probably got to be somewhere in one of these little lists or somewhere. They got to be coming somewhere. I don't know if Brother Mao is busy right now. List of nurses licensed in Tennessee. So they go to board. There's gonna be too much digging right here. I mean, it would be easier if she just came out and showed her credentials and just, you know what I'm saying? If she was going to that extent, because she just made a video and now she gonna have a nursing board come out and then they gonna be like, and then people gonna say they're gonna make a conspiracy out of that. She's supposed to be a nurse in Nashville. All right, so let's see if I can get on the website. I can't see if Malcolm's saying anything yet. But I really don't like doing these shows like this, y'all. Click the nursing board. Okay, hold on. Let me go back to where I was at. Well, I probably could have did it from right there. He says, click nursing's, what he say? Click nursing's for boards. If she ain't a nurse, she ain't got to vaccinate it. All right. Hold on, I'm waiting on Malcolm to say something. Go to the list of nurses. All right. And then click nurses. Click nursings for boards. All right. Now go to registered nurses, advanced practice nurse, licensed practical nurse, medical nurse, nurse aide, registered nurse. Now we got to go to licensed practical nurse. I'm gonna see if he tell me to change it. Default. No, not LPN. All right. Nursing. Okay. Davidson County. Let me know if I need to change it, Malcolm. Yeah, well, we know that she didn't say which vaccination. So we can't even really conclude if she's anything, but we're just dealing with her claim right now. So we should be able to prove that she's a nurse. He says click. What do you say? click the process. Hold on, y'all. I'm gonna make this easy on all of us, cause we gonna find out how to use this website. I send you the link. No county. Y'all jumping the gun. I see my comments being removed. Ain't nobody removing your comments. So if you're talking about somebody removing your comments are not being removed. Corey said, "You dirty, Shawn." Your comments are not being removed. Your comments are not being removed. I am approving comments as they come up on the wall, and nobody's comments have been removed. Don't click county, click Tennessee for location, and then click submit. Okay, all right. If I got all the right stuff up, hold on. Let me see what my commission. Then click process. No, not LPN. Go to default on profession and rank in Tennessee for location. Default to all professions. Default to rank. No county. Don't click county. Click Tennessee for location and click submit. I'm having to approve every one of CK stuff. Do I need to check any of these bottoms? Because it ain't submitting. Oh, it did submit. Is this it right here? Yep. Okay. All right. Okay. So this is the report, and it's in alphabetical order. If y'all see, it says all these things right in here, and then it's got names. Sheesh. Command F. What was her name? Kalila. Let's see. I'm just gonna look by that.
And the average person definitely ain't finna go through all this.
Okay, so her name is Kalila. It wasn't Walker. I think it was something else. I can't remember her name. Mitchell. All right.
Mm-hmm.
And it's going by last name first. And then my computer like, bruh, you can get all this bull crap downloaded. I'm about to delete this.
And Brother Malcolm said he on there.
Yeah, but my computer is thinking right now, so I'm gonna have to hold on a second.
Mm-hmm. Yeah, because ain't nobody reading anyway. Pure Black, you right. They're not finna read that because ain't nobody gonna go and check. If somebody say that they're a nurse and they put on a face mask, that's all Black people need to see.
Yeah,
Unfortunately,
I'm trying to figure out why would she do this.
right? What's her agenda? What's her motives?
Yeah, what purpose does it serve for her to even go to this extent? And now we doing this on a live show, yo. We going in this lady business on a live show. We're trying to go in this lady business on a live show. Yep. Yeah, before attention. I don't know what my computer doing, dude. My computer just really don't do this stuff.
Yeah.
Hold on.
Mm.
Bailey. I do see somebody that I know that's a nurse on this list, though. In them Baileys, 2 people.
You talking about Dr. Mott?
Nah. This is just gonna be in Tennessee.
Oh, okay.
Yeah, this list is all the people in Tennessee. Yeah, man, like the show, share the show. Yup, some more people in here. Like the show, share the show. If my computer wasn't tripping right now, you know what I'm saying? I actually would this stuff that made my computer free. It's trying to like really literally upload all the names. I don't even need all the names. I don't even care about all the names. I just care about one name.
They going in the chat.
Yeah, well, this document is messing up my laptop. Trying to download all that foolishness.
Corey says she put her business out in public. Peace, Zane. I was back and forth, so I'm looking at it now. CK said look up on Facebook.
I can't do anything until it's done.
It's done doing what it's doing, yeah.
Yeah, until it's done doing what it's doing. I don't even know if I can let anybody in. Let me see. Coffee ain't even on real quick. Yeah, I did hit edit fine. Well, I hit command. I got a Mac. I hit command fine, but my command fine, when I hit command fine the first time, I pulled up Mitchell. So after I did that, and then I started scrolling down, it started trying to pull up a new list of names. And so it's trying to load up all that data because this file apparently is huge as hell, and my computer is just sitting here like taking in all these names. But hold on 1 second. Coffee. Let me see if Coffee can boot me real quick, and then I can do something to make it jump back. And then I'll come back. T'Challa, why is doing this? Hold it down for me for a second.
All right, cool. Yeah, man, this is craziness. Everybody want to get online and be seen and heard. Crazy.
Y'all still can see my screen, huh?
Kofi said don't be getting him up out of bed, man.
Nah, don't worry about it.
I'm just playing. I'm just talking.
Coffee. You kick me out.
Zane said look like a Richard sales sheet from here.
Man, bro.
Facts.
What up?
It's crazy.
Kick me out real quick.
All right.
All right.
Peace, Kofi.
What's happening, man? You good?
I'm good, man. You know, Shawn on here doing his thing, chilling with the people.
Yeah. Removed it, eh? Hold on, Shawn. There you go. I removed it there. Let me see. You good, Shawn?
Yeah.
Mm-hmm.
Nah. Don't add me back.
Go
Just
ahead.
remove. Yeah, just remove me, and I'll be right back. I'm finna.
Oh, you trying to see something? Yeah. Hey, peace, Lakeisha. We could see you. We see your comment.
What up with it, Chavis? What up with it, Chavis? Chavis, I
It's
got
Chavis.
something extra for you too, man. In your package, man, I mail off, man. I be trying to look out for people that be supporting us hard, man. So you got something extra in your package, man. You know, peace, sister Ladoja, peace to everybody in the chat, peace to brother Malcolm, um, sister Anisha, brother Zane, as always. Yeah, I looked out for brother Zane too, man. Another good, great supporter, man.
Mm-hmm.
Sure, what Shawn in here talking about science.
Yeah, some lady done got up here talking about she a nurse and her face done went numb from whatever vaccine she say she took. So we just going through. He was pulling up the names of the list in Tennessee, but uh his computer was like, "Yeah, this is too much."
Mm-hmm. Yeah,
Mm-hmm.
boy, there's so much crazy stuff coming out right now, man. Around these vaccine testing, man. I tell you,
Yeah,
that old Shawn right there. Let me add him on back in.
I'm in.
Yeah.
All right, hold on, y'all. Hey, let me get my email off. Yeah. All right. What is stupid ass document at?
brother Morpheus in the building. Peace, brother.
Yeah, I see CK in the building. Peace, CK.
I'm gonna have to delete this too. I'm gonna have to delete this. I don't want no part of this. All right. So here we go, ladies and gentlemen.
Man, we can't see nothing but
Yeah, you're not gonna see nothing.
um,
So Command F, right? Mitchell. I'm in the M, so I ain't even gonna. I ain't. Okay. Let's do Khalil. All right. So I'm going back to the M's. I'm going back to the M's. G H J K L M. We're looking for Mitchell. M I. We in MC. We're looking for M I. Here we go. M I. M I D's. M I L's. M I L L's. M I N's. Mitchells. Here we go, ladies and gentlemen. Here we go, ladies and gentlemen. Now, these contain people's whole names. All right. We have Amy Mitchell, Angela Mitchell, Ashley Mitchell, Barbara Mitchell, Beth Mitchell, Carolyn Mitchell, Carol Ann Mitchell, and then Carolyn Mitchell, Deshay, Donovan Mitchell, Eric Mitchell, Helen Mitchell, Jennifer, Jessica, Karen, Kathy, Lauren, Lori, Jane, Amarella, Margaret, Mary, Megan, Melanie, Rachel. Look, there's two Rachels. There's Rachel with the EAL and then a Rachel with an EL. Susan Michelle Mitchell, Vicki Lynn, Victoria K. Mitchell, Wendy Mitchell, Howard Mitchell, Then we have Katrina Mitchell King. We don't have a Khalila Mitchell at all as a nurse. Now remember, Malcolm made me remove, and Malcolm, I think that's him, made me remove what type of nurse so that we can get all the nurses. This includes LPN, registered nurses, every level of nurse, not practicing nurses, full-time licensed nurses, all of that. This is all anything with the nurse name on it. She ain't in no category. Is that Brother Mal? Or that's Brother Kofi. He dipset. All right, so I did share my screen and y'all did see that, correct?
Mm-hmm.
The extent that we have to go to deal with some of the foolishness in the area is unbeknownst. Let me see if I can delete. Well, what I gotta do to delete all this crap, I forgot. Do I go on here? Nope. Nope. Launchpad? Nope. Y'all got me looking to delete all this food. I'll do this offline. I ain't gonna do this right now. All right, so let me continue, man. Let's get into the bill. So we got sidetracked. Dave sent me on a wild goose chase, ladies and gentlemen. For anybody that's joined in right now, Dave sent me on a wild goose chase because he made me look up a video of a sister saying that she received a vaccination. She didn't say which vaccination that she received. She said that she was a nurse. She didn't list what hospital she worked at. And she made us go there. Nah, it's Khalila. It's Kha. She spelled her name on the video. And I still got the list right here in case I need to go back to it. But let me pull the video up back for the people who just joined so they know what we're dealing with. We're looking for this lady right here. Supposed to be, but look, her name come up for YouTube and it says she's an RN, bro. A registered nurse. Now, what do we know about registered nurses? Registered nurses aren't even getting vaccinated. Are they? Let's see if they received the vaccine. What nurses need to know about COVID-19 vaccine. All right, this is nurse.org. Is that Malcolm? Yep. All right. What nurses need to know. Now, Malcolm, what type of nurse are you?
Registered nurse.
All right,
RN.
so she says she's an RN too. Have you been vaccinated yet?
No, I go either tomorrow or Wednesday.
Okay, so let's do this.
Yeah, and there should be a board of nurses with every state, and they tend to see lists. You know, the list is typically open to the public, and you can verify people's credentials. I think it's the same for doctors too. The difference between a registered nurse and an LPN licensed practitioner, practical nurse is they're not, they don't have the skill set that registered nurses have. They kind of the level below, and they need maybe an additional few years of school to actually get their RN. Okay, your practitioners, they are above nurses.
Last week, USDA federal cleared Pfizer and BioNTech. All right, so if she's a registered nurse, then she would be on that list. And here we have, look, Here we have a registered nurse at Southern Hills right here who received the COVID-19 vaccine. You see her, registered nurse. Now, that's right off Hardin Place. I can tell you that. You see what I'm saying? I can take you there, right? We know Maharim received Moderna. They didn't even get the first vaccines. So where is she a registered nurse at? Here, TriStar Health, nurse practitioner. All right. Tennessee hospitals got 56,000 doses of the Pfizer shot. According to the State Department of Health. You deal with numbers and all of that. Let's deal with it. All right. So what we looking for, HCA, I know where their hospital at. You know what I'm saying? What we looking for, ladies and gentlemen, is that this sister name is she said in the video. Kofi, watch the video. Watch the video, Kofi.
Hi, I am a registered nurse in Nashville, Tennessee, and my name is Khalifa Mitchell. I'm reaching out to everyone about the COVID-19 vaccination. I recently took the COVID-19 vaccination. After the shot, I felt fine, but within 3 days, I went to the doctor because I had problems with my face. The whole left side of my face, actually. I have Bell palsy now. And as you can see, I can't smile. I'm trying to smile. I'm just getting embarrassed. But I just want everyone to know that I think this vaccination is the worst thing ever. And I would not give this to anybody, even my worst enemy. Please, America, they do not care about us. Do not take this vaccination.
Now, they have already administered the vaccine to 2 million people, right? 2 million people, all frontline workers, and they're still doing frontline workers. Hundreds of frontline nurse, doctors, and other healthcare workers from Tennessee Health System's TriStar Health filed into the HCA healthcare building in Nashville on Thursday. All right. Now, on Thursday, let's see when this article was published. December 19th. Let's see when the video comes out. We can't go by that one because this was 6 hours. That was 2 days. This was 2 days. Today's Monday. We don't have any. Let me see. Let's do registered nurse in Nashville, Tennessee. 6 hours, 2 days, 19 hours ago. She said 3 days. So she said she got the shot, and 3 days later she received Bell's palsy. So if this article was published on the 19th, and which means Thursday, Tennessee, Nashville workers start receiving the vaccination Friday, Saturday, Thursday. So we count Friday, Saturday, Sunday, right? So Friday, Saturday, Sunday would be 3 days. So the video would have been available 3 days. Would have came out 3 days. I mean, would have been yesterday. Would have been the first day the video would have dropped. So this would have looked accurate because it would have been literally 3 days. But this video was published 2 days ago by some people. That's the latest of the video. See? Here we go. See? They put it up everywhere. This on some, oh man, I'm gonna get a bug. This was a day ago. 23 hours ago. You know what I'm saying? They running it around. But here's a February disciplinary. They ain't got anything to do with anything right there. So let's see if she.
Now,
go ahead, Malcolm.
I would say now one of the questions I would want to know was she originally licensed in Tennessee? Do we know what she originally was? because some of these nurses are, well, I don't even know. Yeah, some of these nurses are kind of coming to these cities from other places. So I don't know if you got reciprocity here. So if Nashville is the place where she got a nursing license, she is not on that list. But if she's from somewhere else, then it's possible we can look there and see. But she said she a registered nurse though, and said she was in Nashville, Tennessee. So unless she traveling, it ain't real.
So Daryl Blue said, "Why you don't just reach out to the source? Y'all love sources."
Shoot, we going? He you know.
I mean, go ahead.
No. Hold on. Daryl Blue. You should know better than anything else. I'm in Nashville, Tennessee, bro. I should be able to pull up on her and verify if this is accurate or not. She ain't even on the nursing list, bro. So why you just now joining the party? She's not on the nursing list.
And that list is real. My name is on there. You can look it up. It's right there. Right there under Robinson. It ain't fake.
These people ain't even from Nashville, bro.
Nashville, my favorite city, man. I love coming down there. Boy, I come down there, get some of that Manila and
Oh yeah, everybody be tearing my nails up.
I be in there. And my Italian restaurant downtown. Golly, I'm gonna remember that, man.
Yeah, Daryl, I see what you're saying. Like, ain't nobody
Nashville.
trying to attack you, but it was the way when you put y'all. It's the context of communication. Like, depending on what you say and what you say behind that, gives the impression. It implies that, like, you know, because too many people fight against us and be like, "Well, y'all love sources." You know what I'm saying? So it just kind of it appears that way. Reading it online.
I mean, I thought when I went on Facebook that I would be able to find her, but I cannot find her. I'm in Nashville. Shouldn't I have some type of friends that mutual friends to her? Somebody that I know gotta know her. She's not coming up at all. I have a friend that has shared this video on my friends list. Nope, sorry that she ain't on my friends list. I'm looking everywhere, bro. She not showing up on my friends list. Now, out of all the people that I know in my city, somebody gotta know her. And now I'm finna have to go a little bit further. My cousin Sharonda is a nurse. My cousin son Jordan is a nurse. My cousin Keisha just graduated from Vanderbilt, nursing program, right? My uncle Michael, my uncle John, my auntie Alicia, all in the nursing field. That's 6 people. My grandmama, which all them people that I just named, my grandmama was a nurse over the 7th floor or 7. No, she called it 7 Kid Baptist Hospital when it was named Baptist Hospital. They didn't rename Baptist Hospital. My family know a whole bunch of people, so when I got my surgery, I was gonna get the best people regardless. But I can't find anyone. Shawn, try to name in Lithonia, Georgia. I can't find anyone related to this person. I mean, that would even be like that. Oh, let me try Facebook. I mean, all we trying to do is confirm if the sister got it. You know what I'm saying? I mean, something going on with the sister that we seen on the screen, but we really just don't know who it is or what it is or what, anything like that. So all we just trying to do is confirm it. She put it out there. She made it a point to say that she had Bell's palsy. There ain't nothing turning up for me, Pure. From West Africa. Nothing turning up.
I mean, and quite frankly, unfortunately for the sister, it wouldn't change anything for me. You know what I mean?
Right.
I know tons of people who got it right here in Memphis. Like I was just eating dinner with my brother's wife, who was a registered nurse. She got the vaccine. A black lady, I'm at work, with a partner of mine. He got the vaccine in the hospital. Totally fine. Oh man, it was just like getting, wasn't even as bad as the flu shot for him. You know what I mean? I hadn't came across anybody with any issues. But if she have an issue, she can simply send it to the VERS website, the vaccine adverse event tracking system, and they'll get her straight. Put that in a database. It's a very transparent system, unlike any before. I mean, unbelievable how transparent they are about this vaccine program. Right.
Yeah. So yeah, we did.
Believe it or not, it's just what it is.
All right. So for the last 30 minutes, though, we have been on a goose chase regarding this lady. So we know that vaccines from Nashville, we know the rolled out. Now we know that Dr. Hildreth received his vaccination a week ago, right? And we know the nurses began to get on board closer to that. That's why I pulled up that article to show you when the nurses actually start being administered. Now, I guarantee you, post Foster claim that. All right, so I guarantee you that this lady video will appear again with more information, and we're gonna find out because it looked like she lived in the projects in the video, but I ain't judging that. I'm saying we're gonna find out more about this lady because now her name is going to be searched over and over and over until people turn up and can confirm it. All right. So I don't want to harper too much on that. We kind of did get sidetracked, and we blaming Dave for this because Dave the one that brought this bull crap up. Yeah, I'm getting the vaccine too. But I know a brother in the chat asked, have any of y'all got it? I don't know if he was just asking to ask or asking to say what exactly. But Mike Shabazz, Malcolm is in line to get it. And we ain't nowhere near in line to get it. So if you're asking us, have we had the vaccine? That you can't ask us because a few shows ago I showed you a link that shows that there's going to be about a few million people before you, me, and everybody else in the chat. And I don't care what was going on with that. She wouldn't be crying, she'd be paid. Facts. Yep, she would. I don't watch the people that you actually may watch, Daryl. I apologize, but I don't watch those channels. I don't watch misleading or incorrect information. To hear what other people are saying, right? If I'm going to get opposing information, I'm gonna get a credible opposing information so that I can review the data from scientists, not from people in the community who don't have a degree in the field of virology or immunology. So no, that wouldn't be for me. I ain't saying that I'm not stopping nobody from entertaining that. I'm just saying I just went down a rabbit hole because Dave B threw me off. You know what I'm saying? If I listen to Polite, if I listen to Ali Muhammad and all them other people, the pills and so on and so forth, I be in the rabbit hole all day. I can't live my life in a rabbit hole when we got qualified, people out there. It was law, not scientific. Nah, I'm bro, I don't trust him with nothing. I don't trust him with nothing. You remember Cargo Miniums? He supposed to have 90 plus published books and so on and so forth. His history, invalidate him for me. But I'm gonna move on. I'm gonna move on a little bit, man. But does anybody want to say anything else regarding the what Dave sent me on a wild goose chase? I'm blaming Dave for this one, y'all.
No, I just looked at the what's the name? You going in on virology today? A little bit?
Yeah.
All right.
I'm gonna pick out where we left off, and then I'm gonna get into all of that. Oh, my bad, Dave. You're right. Correct. 100 books. No, I'm asking because I work in the hospital and I'm too skeptical to take it. I come in peace with respect. Okay. No. Mike, it's all good, bro. Don't take the response in a illegitimate way. You know, some people who say that, they always tend to say it from respect. So it's respect. They have offered it to me twice and I've turned it down. How much do you know about the vaccine? Yeah, but if she's a CNA, if she's a CNA, if she's a CNA, she wouldn't be in line for the vaccine. If she's training to do anything, she's not in line. She's not she's not on the front line. You have to be registered. You have to be a part of the board to be in, getting those vaccines like that. Okay, Mike, man, what I would highly suggest, brother, is that I'm not here to convince you or anything like that. I would say the first thing that you would do that I would ask you to do, and I know this is probably your first time tuning in, is to, if you got time, like an hour or so, I got videos that'll go back that'll help you understand. But if you don't want to use those videos, in the description of those videos are sourced articles. Get familiar with what the mRNA vaccine is. Get familiar with what the mRNA vaccine is, and then understand how the mRNA vaccine works. And then look at the data. What I mean by the data, we have data. We have the Pfizer trials, we have the Moderna trials. I have all of those in the descriptions of videos as well. And you can see the adverse effects. And by reviewing all the evidence, that gives us an opportunity to come to an informed decision. Now, when you hear us saying that we're going to get the vaccine, that's because we've studied the data. But we're not telling you that you should get the vaccine. We're not making that decision for you or your family. The only thing that we're doing is based on the evidence that we have and based on the climate that we live in. Given all the all of the pros and cons of what's going on, we getting the vaccine. It's just a flat out, and I appreciate your honesty, and I appreciate you even commenting in the chat and saying what you were saying, bro. But I would highly recommend understanding what mRNA vaccines are and what they do. But if you've rightfully declined it twice, I would ask you, based on what understanding or level of understanding that is causing you to refuse the vaccine. Because if you working in the hospital, you frontlining, bro. You seeing this with your own eyes, what COVID-19 is doing to people. You know what I'm saying? And I would just suggest, going back and understanding those things, and then hopefully that will work out for you. Peace, Fitz. I see you in the chat, bro. Man, again, like the show, share the show. I appreciate everybody joining in. There's some new faces in here commenting and things of that nature. Done data my what up? Again, does the vaccine protect from the new strand? We have talked about that. Rob, peace. Well, I'm glad people are filing in right now because we really finna get into some good stuff. But you know what I'm saying? I am seeing people hopping up in this joker. So Daryl, yeah. Yes. In short, yes. They have no doubt that the vaccination would protect against this it's not a new strand, it's a variant. So we have to be clear on that, Daryl. It's not a new strand, it's a variant, and it will protect. We good on that. We're confident on that. Matt, go ahead,
Good
bro.
info. I would say good info, man. Yeah, it's a new variant. It got, new variant and not a new strain. Still SARS-CoV-2. Just has a few mutations somewhere, I think, around the S protein, but still not in the portions where it will affect your body from producing antibodies that will fight it off if you're vaccinated with these upcoming vaccinations.
Yeah, so that's where we at with that, man. I appreciate the questions and comments. But today's show, we gonna deal with viruses, and we gonna deal with the evolution or adaptation of viruses. We picking up where we left off yesterday, pretty much. And I know this might be a little long drawn out. So what I may do is I'm just gonna continue it. Ain't no sense in making y'all do two shows because some people might miss the second show or things of that nature. But I'm gonna get into some of the stuff and then we'll kind of ride out. I mean, we really been on like almost an hour and a half, and we really spent about 45 minutes going down a rabbit hole with a lady who claimed to be a registered nurse at a hospital she didn't identify herself as, but she could have identified herself and says, hey, I'm a registered nurse. Here's my evidence that I'm a registered nurse. I'm not going to tell you what hospital that I work in. However, I received the COVID-19 vaccine, and then said which vaccine she took. Was it Moderna? Was it Pfizer? We need to know. So let's say we find out that she received a vaccine, right? Or whatever. Which one did she receive? Because that is important to us. So the spike protein has mutated. They account for the spike protein mutation in the development of the vaccine. Facts, Corey. Yep, I was just giving them the short end of it, and I got you, Matt. I figured that you had to go back to work, bro. The protein used in the vaccine covers all variants. Correct, Shawn? Yes. Now, if it's a new strand, then they would have to modify the mRNA. Yeah, account it. So again, if it's a new strand, they would have to modify the mRNA, the messenger, the mRNA vaccine. They would have to modify it, which would be faster because they've already been able to map things out and things that they would have to map it out. They would need more data, which means that more testing comes in, gives them more data for the scientists to study, and then they would map it out, and then they would change the molecular structure of the messenger and have it fire off to protect. But yes, account it. Corey on point. But let's get into it, man. Let's get into today's build. We left off yesterday with some stuff, and we didn't get to it. We were talking about virus adaptation, and we were talking about the evolution, right? We got into the ecology and evolution. We were hearing from my dear brother from Yale, and we're gonna continue to pick up the pace with that. So let's see what Mr. Turner has in store for us today. This build is a little, I might be doing a little stop and going. It is also in the description. And then I really want to get to this virology lecture that was given by our Vincent Ransonelli, which was critical. It was given in front of Columbia, and that's about an hour. So, Dave kind of messed us up today when he sent us on a goose chase. But it is what it is. And I'm gonna wait till it come on. Let's go.
Hi, I'm Paul Turner from the Department of Ecology and Evolutionary Biology at Yale University and the Microbiology faculty at Yale School of Medicine. Today, I'd like to present on virus adaptation, or not, to environmental change. This talk describes how viruses have an amazing capacity to adapt to environmental challenges, and yet we'll find that these champions of adaptation sometimes encounter environments that demonstrate that environmental change can constrain evolution and adaptation, and even these so-called champions can face constraint. So very many challenges exist to viruses in the natural world. And you could think of this at all different levels of biological organization. So if you start at the base level of molecules and cells, the primary challenge for viruses is that they cannot control where they exist in the environment. So they might encounter some cell type and successfully enter if they have the right protein binding to recognize a protein on the cell surface. Or they might bump into the wrong type of cell, and that protein recognition doesn't occur. So therefore, it's an immediate and proximate challenge to a virus to infect a cell, depending on where it is in the environment and whether the proper cells exist to infect. In macroorganisms like us, we have tissues composed of different cell types. So if a virus is in your body and it's replicating in one tissue type, it might be challenged to infect a different tissue that's nearby, and it's incapable of doing so. Hosts, such as humans, have elaborate and beautiful immune systems. Some of them are adaptive, meaning that they change through time. And this is a way of our immune system, in a way, keeping pace with microbial invaders and changing at the same pace that they might evolve through evolution. But viruses and other microbes, when they encounter these immune systems, this poses a challenge for them to continue to infect that host or that host's progeny or other susceptible hosts in their environment, depending on whether those immune systems provide an immediate and successful barrier to virus replication. It's an amazing thing that some viruses infect humans and also successfully infect very different organisms that are not at all closely related to us. A great example of this are the arthropods, where many pathogens are vector transmitted by arthropods such as mosquitoes, including viruses. And this is pretty fascinating because a virus has to grow within an invertebrate. And for example, in a mosquito, it has to grow in the midgut and eventually get to the salivary glands in order to be present in a bite that puts the virus in the bloodstream of another host to be picked up by another mosquito. That's got to be an incredible challenge for a virus to grow both successfully in an invertebrate like an arthropod, as well as a vertebrate like you, a human. And last, we have to remember that global level ecosystem changes affect all biological entities on this planet, including the very smallest ones, such as viruses. So when you think about challenges like climate change and global warming, you have to remember that this is something that is felt by all biological entities, and therefore viruses can also feel the challenge of an ever-warming world. There are many different virus study systems that my group examines. So these examples are shown in the very many beautiful forms behind me. On the far left, we have vesicular stomatitis virus, which is an example of a single-stranded RNA virus with a negative sense genome. And in the middle, we have a variety of other viruses also that will infect eukaryotes, but they happen to have positive single-stranded RNA genomes. Closer to where I'm standing, we have single-stranded DNA filamentous phage, and also double-stranded RNA and double-stranded DNA viruses, in this case, both phages, phage 5/6 and phage T2. So these are examples within my laboratory of the wide variety of viruses that exist in the natural world and how a single laboratory can choose to examine this great variety of virus types. Depending on the challenge and the question, we would like to focus on a different study system to examine whether viruses can successfully or not adapt to their environments. A big tool that we use that's very popular with others and a very powerful tool would be experimental evolution. A way to summarize this method is it's the ability to study evolution in action. So if you have the right study system in a controlled place, like a laboratory, you can take that population, put it in an environment that you control explicitly, and then examine how does that population deal with that challenge, both in terms of the traits that it evolves, as well as the genotypic changes that it undergoes. So both phenotype and genotype can be the focus of these studies. An important thing to remember is even if a researcher is manipulating the environment in the laboratory, it still can be a challenge to a population, and that population can evolve through natural selection. So you're talking about an artificial environment, and yet natural selection can occur. That's because the researcher is not determining which variants in that population will better match the environment. Instead, that's due entirely to the mutations and the genetics of that system to meet that challenge or not. And that can happen through the process of evolution by natural selection. A typical design is shown here, where we would begin with some ancestral type. We might be interested, in the case of this hypothetical diagram, in 3 different treatments that differ in some way in their environmental challenge. And we can track over the course of generations, how do these independent lineages evolve to meet these challenges? And the nice thing is to include replication in these experiments, such as you can have lineages that are experiencing the same environment. And you can look at how consistently do lineages undergo random mutation, and yet the same mutations might be the ones that rise to fixation and lead to adaptation. In other cases, there might be different solutions to the environmental challenge, and you'll see divergence between your lineages in the sense of different mutations are meeting the same challenge. Another way to think about these experimental evolution studies is to create a hypothetical diagram of some phenotypic trait that you would want to measure. This might be growth or some other capacity of the system to meet the challenge. So in this example, I'm illustrating how this trait has some variation at the beginning, and then we would create some sort of an ecological circumstance or an environmental challenge in these studies. And through time, we can keep track of how phenotypes change. So you'll notice that the average phenotype along the x-axis in this hypothetical example is shifting to the right, meaning that the mean of the distribution is changing according to which variants are in that population and the ones that are best meeting that challenge. Now, we can go further than a lot of systems because it's very easy for us in virus studies to take the entire genome from these evolving populations and explicitly look everywhere in the genome for where a mutation might occur. In this case, we can track through generational time how is the genetics changing in relation to the ecological challenge as well. And this allows a lot of immediate power in making something called a phenotype-genotype association. You can infer how the changing phenotype is being controlled by underlying genetics and make some base inferences about what the relationship is. And I don't want to trivialize that because one has to do a lot more work to convince oneself that perhaps one mutation is responsible, maybe 2 or 3, or even more complex things can occur, like these mutations acting with one another through properties like epistasis. So this provides an amazing amount of power to examine how evolution occurs according to the environment that you create in these types of studies. The outline for what I want to talk about today is pretty much centered on these 2 questions. We can consider environmental changes as fostering versus constraining virus adaptation, depending on how the environment is constructed in these types of experiments or in the natural world. And especially now, that takes us to this next question of, are there particular traits that can evolve in viruses that match something intriguing that you see in cellular systems? The investment in survival versus reproduction is often something that's seen as at odds to one another in cellular systems, that you can either invest a lot in survival as an evolutionary trait, but this minimizes your reproductive capacity, or vice versa. So an intriguing set of studies show that this same constraint or this same trade-off can happen even in non-metabolizing organisms, such as the viruses, especially in the viruses. How does environmental change foster versus constrain virus adaptation? Let's look at this question first. Virus emergence is an amazing biomedical challenge that we face today. So even though RNA viruses especially are not that prevalent among the highly prevalent viruses that exist on this planet, they seem to be especially able to jump into new host species and cause harm through disease. So humans see this through recently emerging pathogens, such as Zika virus, which is sweeping around the globe and is problematic and creating a challenge to biomedicine to protect people against Zika virus infection that can disrupt normal development at an early age. A very different example, but still called emergence, is when a virus comes from another species, enters into the human population, and gets locked in and becomes very specific to humans. So I began with the case of Zika virus, which is not specific to humans, but a great example of a specificity evolution would be HIV, which came into the human population several times independently from our primate relatives, especially chimpanzees and certain species of monkeys, And this has led to the evolution of HIV-1 and HIV-2 independently several times. A third example of emergence would be something that exists both within humans as well as in other species. And a great example of that would be influenza virus. So ordinarily in any year, you can have plenty of the human population seeing flu virus infection and suffering influenza. But what we fear is that there are certain forms or genotypes of influenza virus that will be especially virulent and cause a high degree of mortality and sweep around the globe to infect a lot of humans and adversely affect human populations more than a standard flu season. And especially we fear that the large reservoir of influenza viruses that mostly exist in this planet in waterfowl might lead to a variant that can jump immediately into humans and they be passed from human to human. This would be an example of a flu virus coming from a bird, coming into a very different host species, a mammal, and causing a lot of destruction and mortality because of the inability of the human immune system to deal with the challenge. So these are three different examples of the same cataloged thing. That thing is what's called emergence. And this is a huge biomedical challenge. We can think of how emergence can or cannot occur for a virus, and that's what I want to focus on next. And there are some certain fundamental expectations if you have any lineage, whether it's a virus or not, and whether it's encountering an environment that is constant through time versus changing seasonally or in a temporal way through time. So I'm giving two hypothetical examples of this. At the top, we have a hypothetical evolving lineage that sees niche A and niche B in a flip-flopping fashion. And each one of these little circles indicates a generation. So necessarily, this lineage has to grow in environment A in order to make it long enough in its environment to reach a new environment B, and so on. Necessarily, we would expect that this will select for generalization, the ability to thrive in both of these environments, because there is no other option. Now, that's very different than if that lineage has the luxury of seeing only a single environment. In this case, environment A is the only thing it encounters, but I'm underlining the word tends to select for specialization because that's only one possibility. This luxury affords this possibility of being highly specific to your environment and being very good in that environment. But it also is an opportunity for generalization to occur if you have a correlated response to growing well in other environments. And essentially, that must be happening in emerging virus pathogens. They happen to have the right genetic capacity that when they jump into a new host species like human, they can just really hit the ground running and grow very well, cause a lot of damage, and ultimately they might be specific to that environment, ultimately, but initially they're highly generalized. We've covered this topic in a variety of papers that I'm listing here that I won't have time to go into much detail, but one can think of this challenge of virus specialism versus generalism happening a lot in the natural world, and it's very easy and powerful to study this in the laboratory through the experimental evolution method that I mentioned earlier. One system that we focused on a lot to study how virus specialization versus generalization and just simply adaptation can happen is a model known as vesicular stomatitis virus. So this is a single-stranded RNA virus with a negative sense genome that's pretty much a workhorse in molecular virology. It's been used for very many decades to understand fundamentals of how RNA viruses infect and replicate in a cell. So
I hope y'all just caught that. Because what we dealing with right now,
the negative sense genome, that's pretty much a workhorse in molecular virology. It's been used for very many decades to understand fundamentals of how RNA viruses infect and replicate in a cell. So some pictures here that I'm showing are just to reflect that we have a lot of prior knowledge for the molecular details of this system. And that's great when you enter into experimental evolution studies because you don't have to go about measuring that stuff all over again. You can think of the outcome of your experiments in the context of the prior knowledge. So VSV has a very small genome in size. It has only 11 kilobases in length. And this comprises only 5 genes. So one could think of this as a pretty simple system. And yet it has a pretty amazing capacity to do things like both reproduce in an arthropod. It's an arthropod-borne virus or an arbovirus, and it also can replicate in a mammal. So in the case of VSV, it's a safe system to use in the laboratory because it might get in a human by accident, but it really doesn't cause much harm. It's agriculturally important in large mammals, domesticated horses, etc. So we do care about it from a disease standpoint, but it's a great, powerful system to use in the laboratory safely. It comes from the family Rhabdoviridae, which also features rabies virus. Here's a summary of some of the data from one of our experiments where we harnessed experimental evolution to examine how does this virus deal with a constant environment versus one that is changing through time in that temporal heterogeneous way that I outlined. So this is a pretty busy diagram, but I'll walk you through it. At the top, this is merely a depiction of the VSV genome and the 5 genes NPMGL. And what you can see is for each one of the lineages, the 4 lineages that saw only, in this case, HeLa cells, those are cancer-derived cells that originally came from Henrietta Lacks a long time ago. And these were harnessed as an immortalized cell line that people use in a lot of studies beyond simply virus studies. But these cancer-derived cells provided a new challenge for VSV in this experiment. And each one of the points here on the diagram are showing where these lineages changed in their genetic material relative to the ancestor after the experiment took place. In this way, we can catalog what are the mutations that arose and which ones fixed through natural selection to let these lineages improve in their environment. We also did in this experiment a challenge where the viruses had to not only evolve on HeLa cells, but in an alternating fashion, they had to enter into a non-cancer derived cell type, abbreviated as MDCK, and in this way they had to become adapted to both HeLa cells as well as these non-cancer cells. And you'll see that we also cataloged their genetic changes through time. And this has a great deal of variety even within each treatment for the mutations that fixed according to each lineage. One can also catalog the exact position where each one of these mutations took place. Let me highlight one more thing before I move on, and that is really these virus populations after this experiment are not carbon copies of one another. So there are many places where we do see that they underwent the same mutational change at exactly the same place, and that must be evidence of some beneficial mutation coming in and fixing in these lineages. And yet, in some genes, they underwent different mutations from even the same populations in the same treatment. So this indicates that there can be other genetic solutions to the same environmental problem in a study like this. Keep this in mind as we go on and look at a subsequent experiment that challenged the ability of these viruses to evolve and infect yet new hosts to test what is their emergence capacity. Simply remember that we lump them together as specialists having seen only one constant host type, or generalists that were selected to see 2 types, and yet the lineages are not carbon copies of one another when they're drawn from each treatment.
All right, so I'm gonna stop him there. You know what I mean? And I'm gonna let that marinate on y'all a little bit. That was a lot to digest. It was some good build in the conversation. I want to get into Vince. I want to get into his build because it's going to also shed light on what the Professor Yale was actually building on, and this one's going to be a little bit, you know, around that lane. Yeah, Racinello.
Welcome back. Today, we're going to talk about emerging viruses. What is an emerging virus? Well, we define it as the causative agent of a new or previously unrecognized infection. So you can have a brand new virus that you've never seen before, or a virus you've known causing something different, or a virus that's been there and suddenly you're recognizing it. This term was introduced in the 90s, but of course, emerging viruses are not anything new. Ever since the rise of agriculture, new infectious agents have invaded human populations. Why? Because the populations have become large enough to sustain infectious agent transmission. Before agriculture and commerce, humans were hunter-gatherers. They lived in small communities, didn't support much infection. Certainly there were infections introduced into those small communities. They didn't typically spread beyond them, and no one, of course, knew about them. And this idea of an emerging virus became popularized, became something to investigate because we got the technology to detect these emerging viruses. And of course, the popular press is very interested in emerging viruses, as witnessed by this 1995 Newsweek cover on Ebola virus. An emerging virus can be a virus with an expanded host range or an increase in disease that we didn't previously notice. So, for example, Zika virus was discovered in 1947. However, it wasn't until 2015 that we recognized that that mosquito-transmitted flavivirus could cross the placenta and cause microcephaly and other congenital birth defects. Now, we have a term embedded in the idea of emerging viruses that is zoonosis. A zoonosis is the transmission of a virus from a wild or domesticated animal to humans. We'll use this many times today. You'll get to understand it a little better. SARS-CoV-2 was a zoonosis at one time. It came from a non-human animal to a human, but now it's no longer a zoonotic virus. It is a human virus. Sometimes a zoonotic infection or a cross-species infection, sometimes we call it a spillover from a non-human to a human. Sometimes this establishes a new virus in the population. Simian immunodeficiency virus, for example, moving from chimps to humans in the 1920s. We'll talk about that in its own lecture. SIV was not a human virus. When it infected the first humans, it was a zoonotic infection. Then it became a human virus. And of course, SARS-CoV-2, as I've just said. Now, in contrast to these zoonotic spillovers that eventually become human viruses, often cross-species infection can't be sustained. For example, Ebola viruses continuously spill over from bats to humans, but the chains of transmission eventually end, and the next outbreak is a brand new spillover. Same thing with MERS coronavirus from camels to human. Every infection is a brand new spillover. Same thing with West Nile virus. Every time a mosquito transmits it to human, it's a spillover from a bird to human. And this book, Fever, about Lassa virus, Lassa infections continue to this day. Each one is a spillover from a rodent reservoir to a human. Each one is a spillover. It doesn't go from person to person. Yet there's so many initial spillovers that it's a problem. Every human virus that we have today originated in a non-human source. We mentioned this last time. I want to emphasize this again with this graphic where we're looking on the y-axis at the viruses belonging to the number of genera. These numbers here are genera that harbor viruses of different sorts. First on the left, 31. Viruses belonging to 31 genera are human viruses. This includes measles, herpes viruses, mumps virus, rhinoviruses. All right, these are human viruses. They once originated in animals, but now they're human viruses. Viruses belonging to 37 genera are zoonotic pathogens. They spill over from zoonotic sources, like MERS coronavirus, like Lassa virus, like Ebola virus, Nipah, Hendra. They're all brand new spillovers. They don't transmit very well from human to human. And then the remainder, 16 viruses belonging to 16 genera, we inherited. Homo sapiens inherited from its hominin ancestors, like Australopithecus had a virus that was transmitted down through all the descendants eventually to Homo sapiens. So 16 of those. And then finally, 6 came from Homo species, like Homo neanderthalensis. So that's the ancestral sources. Now, these last numbers here, these primates, of course, obtain them from animals. So it's not like they developed in them. They're all from non-human animals. The biggest overriding factor that drives the emergence of infectious diseases, not just of us, but of animals as well, is human population growth, as evidenced by this amazing graphic where it's just shot into the billions quite recently. And of course, accompanying this population growth, amazing change occurring in all ecosystems as we inhabit every corner of the planet. And we introduce changes that alter the ecology. We'll talk more about ecological issues next time. And we make encounters with viruses more and more frequent. And these are some of the ecological and anthropogenic activities that promote virus emergence. Another way to say this is humans continuously devise new ways to encounter viruses from our hunter-gatherer ancestors. They lived in very small groups of 100 or 200 people that moved around when the food ran out. They encountered some wildlife and got perhaps infections from them, to now we have amazing global travel networks. This is a website where you can see all the airplanes in the air at any given time. A virus can go around the world very quickly. We also do other things that increase our ability to become infected with viruses that were previously not in us. Dams, water impoundments have led to so many new viruses emerging as the dam creates new environments for different hosts of viruses. Deforestation, rerouting, migration patterns, wildlife park, long distance transport of livestock and birds. People want to buy exotic pets. Wow, they come with their own viruses. Blood transfusion, xenotransplantation, sexually transmitted viruses, drug abuse. And of course, there's air travel, urbanization, daycare centers is a recently devised invention, which is a good way to get lots of kids together and share their viruses, then bring them home to their parents. Hot tubs, improperly sanitized, spreading viruses, air conditioning, and millions of used tires, of course, which we talked about, transporting little bits of water to harbor mosquitoes, and climate change, and even natural disasters alter the viruses that are around. Climate change, of course, changing ecosystems and increasing the spread of mosquitoes, just for a few examples there. So these are the ways that we have devised to encounter new viruses. But that's not enough. There are two big components of emergence. One is encounter. The other is the genetic diversity. Here's an example of both. This is the Amazon North region of Brazil. Okay, South America, Brazil, Amazon North expanded here. And these are some of the viruses that we know inhabit animals in this area. Many more than 183 arthropod-borne and other vertebrate viruses. Some of them have been isolated. You can see the dengue virus, 4 serotypes in different places here, but also many other. These are all virus names, exotic virus names. And some of these have been isolated. What does this mean? Well, there's a huge diversity of viruses living in wildlife in parts of the world. And of course, the Amazon North region is being deforested. So people are going in here to work. They're going to get their viruses and bring them back into the cities and towns. And so the more we encroach upon these infected animals, the more we're going to get their infections. That's just one example where we happen to know a lot of the
I want to stop right here at the 10-minute mark, because yesterday we talked about arboviruses. And there was a lady on the panel that talked about going into Kenya and doing work into Kenya. Now, today we have Brother Vincent in his presentation that he's done before. This is the second time he's actually done this presentation. He's talking about going into like the Congo regions or into Africa, in the untouched areas where mosquitoes dwell. Now, I talked about the arbovirus for a reason, because mosquitoes are dwelling here. They are coming into contact with, animals who could be infected with something that they have encountered in this forestry area. And then you have humans coming into the area that are working in the area as well, who come in contact with the mosquito, or i.e. the animal in which they have contacted and could possibly be killing, hunting, cooking, and eating. Right? So, you just can't think of it in the perspective of Africa. You can go to Louisiana and you can catch people eating possum. Um, you can go, well, what some people call gopher rats. Um, of course, people eat alligator, lizards, all kind of like weird type of animals and all of that things. And that's a way that also to transmit a virus by eating wildlife, certain wildlife that live in areas that are near wetlands and forestry that tend to be, intermingling with all different types of environmental issues that could be an issue for us over the long run.
Viruses that are present. Here's a list of some emerging infections that we recognize and some of the factors that lead to emergence, like dengue viruses, any conditions favoring mosquito breeding, like tires, and also population growth. Ebola virus, contact with an unknown natural host, possibly bats, perhaps non-human primates. Hantaviruses, agriculture and human rodent contact during the rice harvest in many Asian countries that allows viruses to be transmitted from the rodents. Hendra virus, proximity of fruit bats near stables, for example. We'll talk about that today. HIV-1, hunting and butchering of infected primates or bushmeat trade. Influenza virus, its reservoir in aquatic birds and the expansion of bird and pig farming, which are hosts for these viruses. Middle East respiratory syndrome, coronavirus, camel husbandry, and their contact with humans. Nipah virus, again, the proximity of fruit bats to piggeries. SARS, classic SARS, number 1, open air meat markets. Sin nombre virus, driven by weather changes, which cause increasing in the numbers of deer mice and subsequent contact with humans. West Nile, mosquito transmission from a bird reservoir and global travel, bringing it to the US from Israel. In 1999, Zika virus, mosquitoes and global travel. And now we have SARS-CoV-2. What factor led to its emergence? We're not really sure yet. We have to see where it came from. It likely came originally from a bat, but how it got into people, we don't know. We'll have more to talk about that later. All right, so there's the one role on the one hand of humans making opportunities to encounter new viruses. And most people don't think about this. They go about their daily lives. They don't even think that they're going to be encounter viruses yet. They emerge and they have huge impacts, as we can see now. At some point, we're going to have to do something about this. The other factor, of course, is evolution. And remember, last time we talked about the enormous diversity present in a virus quasi-species. Many of these emerging viruses are RNA viruses. Populations are incredibly diverse. And so you have various non-human animal species harboring their own viruses in huge numbers. And somewhere in that diversity of viruses in those animals, there's a virus that could infect a human, perhaps, just randomly arising by the enormous diversity of mutation that occurs during replication. So the quasi-species, incredibly important in driving this. And I want to emphasize, this is a completely random event. The mutants arise. There is no goal on the part of the virus. The mutants are just arising, and they happen to, say, bind a human receptor and allow entry into human cells, and then selection takes over. It's completely random. Now, our travel, our anthropogenic activities is not random that allow us to encounter animals. We could control that, but this we can't control. And this happens randomly because viruses need this diversity, as we talked about last time. Before we go into some examples of emerging infections, let's talk about the general categories of the interactions between hosts and their viruses that we know about. There are 4 I want to talk about.
Hold on one second.
They're shown here on this graph and how they relate to one another on this graphic. We have stable virus-host relationships. Here in green, this is what maintains the virus in an ecosystem. In humans, think of herpes viruses or measles viruses or rhinoviruses, many, many others. They're human viruses. They are stable in us, and we transmit it to other humans. We could transmit those viruses to other species, which might lead to some of these other categories, like evolving virus-host relationships. This is when viruses pass to a naive population, an immunologically naive population that has never seen that virus before. It could be other humans, could be a group of humans living somewhere that's never encountered the virus that you have, or it could be a different species entirely. And it can go from animals to us. The stable doesn't have to be in humans. It could be in bats, and they give us their viruses. So that's evolving relationship, because once the virus comes into a new host, it evolves for some time before it may stabilize. And these are where many emerging infections come from. Then we have dead-end infections, a one-way passage to a different species. So this can be from humans to non-humans or vice versa. The virus enters a different host where it's not normally, and that's it. May replicate in that host, may or may not make it sick. It's not transmitted. End of story. Dead end. It's quite clear. And finally, a resistant host where there's no infection whatsoever, an insect virus passing through our intestines, right? When we eat cabbage that has insect viruses on it, we're a resistant host for that virus. Nothing happens, just passes through us. These kinds of resistance encounters can come from stable or evolving interactions. All right, so let's look at some of these in a little more detail. Let's look at the stable host virus interactions. These are where both participants survive and multiply. The virus proliferates in the host without wiping out the host, because the host is needed, of course, and the host evolves to be able to deal with the virus. And some of these are very permanent, as you might gather. We've already mentioned measles virus, herpes simplex viruses, smallpox viruses. We're the only host, and the virus exists in us. We've been able to eradicate smallpox simply by vaccination campaigns because we're the only host for the virus. It's a good example of what happens when the virus no longer has a host because of immunity. It's gone. And sometimes these stable interactions may include infection of more than one species. So influenza viruses goes among humans and birds and pigs, for example. Flavi and toga viruses, all multi-host stable interactions. And again, these stable interactions can lead to resistance, evolving, and dead-end interactions as well. Then we have the evolving host-virus relationship here in yellow. The hallmarks: instability, unpredictability. And, you can go from a stable to an evolving interaction, even from a dead end to an evolving, but more rarely. The major pathways here are in red. And this can have any range of outcome from benign to death. So we have some examples here. Smallpox and measles viruses introduced to natives of the Americas by old world colonists and slave traders wiped them out because they'd never seen it before. They had zero population immunity because of the effect of barriers to travel in those days, which don't exist anymore. West Nile coming to the Western Hemisphere in 1999, highly evolving and unstable relationship until it spread across the country and had established infection for a few years in native birds, of course, not in people. And the introduction of myxoma virus to eliminate rabbits into Australia. Very different outcomes in the first year versus the subsequent year. So that's what we mean by instability and unpredictability. And then we have our dead end interaction, which is a very frequent outcome of a zoonotic spillover, a cross-species infection. This happens often, very often, far more than we know. We have a stable virus-host relationship, and the virus spills over into a dead-end host. There is typically no sustained transmission from these new infected hosts to others of the same species. No sustained transmission. That's the key. It can transmit. You can have short chains of infections, but they're never becoming a human virus. They never become a stable interaction in this new virus host system. Ebola virus is a good example of this. Dead-end hosts are humans, chimps, and gorillas. We believe these viruses are acquired from bats, and we'll talk about that in a moment. And the chains of infection are typically not very long, although in 2015 it was an unusual situation and there were some extended chains of infection. But typically these all end, and then the next outbreak is a new zoonotic spillover. Influenza H5N1 virus, highly pathogenic avian influenza viruses found in birds globally and rarely come into humans, but very, very short chains of infection, dead-end infections. These contribute little to the spread of the natural infection. It is the stable and, to a lesser extent, the evolving relationships that contribute to the spread of viruses in nature. And here are some examples of stable and dead-end host virus relationships using two different virus host systems. Here we have a virus which is spread by mosquitoes among wild birds. So this mosquito bites a wild bird and picks up the virus and can deliver it to another of the same species in a cycle from bird to bird. That could be West Nile virus. Sometimes the mosquitoes might bite domesticated birds like chickens, and sometimes other mosquito species can take a blood meal on this particular host and spread the virus to other hosts as well. So this is a stable host-virus relationship. Typically, the hosts are fine with the virus. They don't get sick. These are long-standing infections that have evolved for many, many years. In some cases, these mosquitoes may bite other hosts that are not part of this stable host virus cycle. For example, these are arboviruses that can be transmitted to horses and cause encephalitis or even humans. These are dead-end hosts. Although these animals may get sick, they don't all get sick. The infection can range from benign to serious. They don't produce enough virus so that if a mosquito bit them, it would be transmitted to someone else. These are dead-end hosts. They're not part of the natural host virus cycle. So that's in one example with a mosquito-borne virus. Here's another one with a tick-borne virus, just for some variety. Tick-borne encephalitis virus, which is involved in a stable host virus relationship with rodents. So the ticks pick up virus when taking a blood meal from a rodent, can spread it to another rodent. The mice get infected. They don't get sick typically. They can give a blood meal to another tick and the infection will be perpetuated in this tick-rodent cycle. And these viruses can also spread transovarially in ticks. So the eggs of a female tick can harbor the virus. And when the female lays eggs and they hatch into new ticks, or they develop into new ticks, the ticks will have the virus. And those ticks can go on and bite other rodents. Occasionally, the tick will bite a human, transmit the infection, can range from benign to serious. But again, the human is a dead-end host. Humans can also acquire the infection by drinking milk from another dead-end host. The cow that's been bitten by a tick develops an infection. The virus goes in the milk. And if you drink it, you can get infected. But again, you're a dead-end host. You don't spread it to anyone else. So that really nicely illustrates both stable and dead-end relationships. Now let's talk in general about emerging infections before we go into some specific examples. There are two steps involved for the emergence of a new virus infection. Obviously, the first is the introduction. So a human has to encounter a non-human animal that has a virus in it. And those animals, as we said, have enormous genetic diversity in their viromes. And so perhaps in that diversity is one that's able to infect the human. So that's the key. You have genetic diversity of the virus and you have an encounter. You have to have an encounter. If there's no encounter, you will never see the infection. And that's why we have more and more encounters these days, because our population is growing incredibly. We're all over the place and making all sorts of changes of the types that we mentioned. All right, so that's the introduction. But then the infection has to become established and disseminate in the human population. It doesn't help if the virus infects one people and that's the end. It will never spread. Well, it does help us, of course. We don't want that to spread. But for the virus, it's not going anywhere. That's the end of the infection. However, the virus exists in its natural host, so it doesn't really matter. You can see there's all accidents. These are all accidents with the exception of us building the encounters. So 2 steps: introduction, establishment, and dissemination. Most of the time, as I said, these rare encounters of viruses with new hosts, we never detect them. I'm sure they happen way more frequently than we know, but we don't try to detect them. We don't try and detect every virus that we encounter all the time. And some of us are encountering more than others, depending on where we live, for example. But most of them never go anywhere. In fact, 99.999% of these chance encounters never go anywhere. You may have infection of a single host, so the virus may never even replicate in the host, but it may replicate in that host. It might not be transmitted. It might not replicate to high enough titers. Maybe it's not well adapted. It will not be shed. It won't be transmitted to others. And we will tell the story of how SIV went from a chimp to a human and became HIV. But it's thought that there were many spillovers of SIV from chimps into humans over the years, and most of them didn't go anywhere. And a few conditions were met, the perfect storm, if you will, that led to the emergence of HIV. And of course, horses, pigs, chickens, bats, they can all be sources of viruses that we encounter. And mosquitoes and other biting insects may do the same. The successful encounters, the ones that lead to further replication, of course, the virus has to have access to both susceptible and permissive tissues and cells. If it doesn't, that will be the end of the infection. For transmission to a larger population density and health, the overall health of the population are important factors, of course. If a farmer in a field picks up a virus, it may infect him and his family, but if that family is alone and doesn't travel much, it will never go anywhere else. And there are probably many, many examples of such localized infections. In the end, the virus will endure only if it causes serial infections or chains of transmission. And most animal viruses, non-human animal viruses, are simply not able to sustain these chains of transmission. We know this because we see examples in Ebola viruses, West Nile virus, MERS coronavirus. We know that chains of transmission don't occur. And what allows viruses to make these chains of transmission? What are the changes that go from a rodent virus to a serially transmitted human virus? We have no idea, because often we don't have the virus just before it's able to serially transmit in humans. It would be very nice to have those, but we need to do a lot more wildlife surveillance than we're doing these days. So let's talk about some examples of viral emergence. And here are two viruses that are well known to humans that originated in non-human animals. Smallpox virus, the virus that's been eradicated. If we look at the genomes of all the pox viruses that we know of, including smallpox viruses and all the others that infect many other animals on the planet, and we look at the speciation of their hosts, we think that this virus emerged 3,000 to 4,000 years ago in Eastern Africa, in the red circle here. And we think it was transmitted to humans from camels. Now, camels were introduced to Africa 3,500 to 4,500 years ago. They originated in North America, and they were brought south and across the land bridge into Asia and the African region. And so we think these camels were brought here. They encountered gerbils, a rodent here, which occupy this hatched area of the middle region of Africa here. We think that camels were infected with a smallpox ancestor from gerbils, and then the camels passed it on to humans. And we can deduce this by all the phylogenies that have been done. So that's a smallpox virus. Again, came from an animal. It didn't arise in humans. Measles virus, a similar story. Measles virus is very closely related to a virus of cows called rinderpest virus. And we think measles virus of humans, human virus, it's only a human virus, probably evolved from an ancestral rinderpest when humans first began to domesticate cattle in the 11th to 12th century. So we started the transition from hunter-gatherers to people living in larger and larger collections. And that was made possible by agriculture to feed everyone and by raising animals like cows for milk and for meat. Started raising animals like cows, we got their viruses. They had their viruses long before humans. And we think measles established in the Middle East when human populations began to congregate in cities, cities only made possible by farming, by agriculture, to have the food to feed those cities. Because remember, before we were hunter-gatherers, we went out and got our food. If you're now living in a big city population, you can't go out and get your food anymore. Too much competition, it's too far away, etc. So you need farming. And measles maintenance requires very large populations. So this could only happen when the numbers got to those levels. And then it emerged in the Middle East and was spread around the world by colonization and migration, reached the Americas in the 16th century, and essentially destroyed many Native Americans who had never seen it before. Again, it rose in the Middle East, and it was transported around the world. And that leads us to diseases of exploration and colonization, which are really good examples of how the introduction of a new virus into a naive population can have devastating consequences. And this is a human virus. So this is smallpox and measles, but here we're focusing on smallpox. And we're looking at how when you introduce a virus into a naive population, devastating consequences. So smallpox reached Europe first from the Far East. Remember, we said it emerged in Eastern Africa, in camels. It then reached Europe, started causing big epidemics because lots of concentrated numbers of people. And then it was brought over to the New World. It was actually imported into the Aztec region from Hispaniola in 1520. And it killed 3.5 million Aztecs. And that's why Cortez was able to conquer the Aztecs, because he had no military prowess. They were all sick. Most of them were dead. And so this changed the balance of human populations. Viruses have changed history. We seem to be changing it, in fact, today as we speak. So examples of introduction of a new virus into the same species, humans to humans in this case. Then there are changes in human populations and environments that allow virus spread. And I think a wonderful example of this is poliomyelitis. Poliomyelitis has been recorded since ages ago, thousands of years ago. This Egyptian carving from 1500 BC shows a priest with a withered leg with a drop foot. That's exactly what polio does. And there are all sorts of records in history of sporadic cases of polio for 4,000 years. So this is a virus that entered humans a long time ago. We don't know what the source was. And it entered into a stable virus-host relationship and just caused a case here, a case there. There are probably many, many cases of infection, right? Because we know that paralysis is only 1 in 100 infections. So there are lots of infections, lots of spread, fecal-oral spread of this virus. But suddenly, at the beginning of the 1900s, this virus started to cause epidemics of bigger and bigger size, until in the US alone, 20 to 30,000 cases. And of course, we developed vaccines to deal with this. But it changed. What changed in the beginning of the 20th century that made this from a sporadic disease to an epidemic one? This would be called today an emerging virus infection. The pattern is completely different. Even though it's the same disease, it went from sporadic to epidemic. Well, this is a disease of modern sanitation. And here's the idea. Before 1900, the virus circulated freely. When you were born, you got infected. There were feces everywhere. You had contact immediately with contaminated feces. You got poliovirus. But guess what? Your maternal antibodies prevented you from getting paralysis. You get a mild infection, you develop lifelong immunity, and that was the end of it. But what happened? Well, around 1900, the greatest invention of mankind ever, the toilet and sewers. That kept the feces off the streets. Now, infection is delayed until later in life, and now you have thousands and thousands of kids that didn't encounter feces early in their life. They're susceptible to polio. The virus is introduced into them. Boom, epidemic polio. Very simple explanation. A delayed infection because of modern sanitation. If there had been genome sequencing in 1908, Would the emergence of epidemic polio been blamed on mutation? Absolutely. That's all we do today. Whenever a virus changes in some way, we blame the genome without looking elsewhere because it's a little harder to look elsewhere. These kinds of studies are difficult. It's too easy to blame the genome. And I can guarantee you that every property is not just the genome, but it's something else like this. That's why this is a great example. Another example of environmental changes that lead to virus emergence. Here's a good one where the changing climate leads to changes in animal populations, and that causes infections of humans. And this is hantavirus pulmonary syndrome, a very serious respiratory disease, first noted in the Four Corners area here in 1993, where these 4 states come together. There's a place called Four Corners. There's actually a plaque. You can go step on it if you want. And this disease emerged around there. These are young people who were getting severe and lethal respiratory disease. Nobody had any clue where it was coming from. Turned out to be caused by a virus, which eventually got the name C. nombre virus. Turned out to be endemic in the deer mouse, Paramiscus maniculatus. These are just mice running around the woods. People started looking. They found the virus in them, the same virus that had caused the disease in humans. And it turns out if you catch these mice randomly, 30% of them are virus positive. Now, this virus, C. nombre, was originally called Muerto Canyon because that's the name of the area where the original cases were, but the residents didn't like having a virus named after their town. So the CDC went through a couple of names and they didn't like any of them. Finally, they just said C. nombre, which I think is a wonderful way to say, fine, you don't want a name? We'll give it no name. Anyway, this turns out to be a bunya virus. It's an enveloped RNA virus with 3 single-stranded RNA segments. And these are present all over the world. There are human versions.
If y'all are not catching the theme of this right here, when he gets to talking about what type, the molecular breakdown of these particular viruses, a lot of these viruses are RNA viruses. This is why we need to get familiar with what RNA actually is. What is he really saying? What is he really saying? There's something in these viruses that allow them to attach to us and invade us and then travel and spread inside us, infecting us, causing illness. And we're missing that point. We're missing how these viruses are acting. We've been saying that virus has been here for billions and millions of years. I put B first before M. Billions and millions. And it's evident that, and we really need to know how these things are behaving because RNA, this is not new to them. They have been studying this for a while to understand, hey, these viruses are RNA. Okay, so how are we going to counter them? We're going to work on an RNA vaccine one day and bring it to the forefront to put its hands on the next pandemic. They have been preparing. They have been preparing to create a messenger RNA vaccine that can train the body to create the spike protein to defend itself against the S protein, to defend itself against COVID-19. So when it encounters COVID-19, it already has developed an immune response to defend itself.
And there are mouse versions and many others as well. Why did this emerge, though? What was the reason? These viruses have always been in mice, but why this year? In 1992, there was abundant rainfall, more than usual, and this resulted in a large crop of pinon nuts, which people and mice like to eat. So the mice population rose in numbers, and that made it more likely that they would have contact with humans. They go in humans' houses, they nest under the floor, and the virus is excreted in their feces. And so the mouse feces contaminates blankets or dust. if you have mouse feces, they dry out, and then you go and you vacuum it or brush it up, you aerosolize the virus, you inhale it, and then the virus infects your lungs. So more mice, more mouse feces, more opportunities for human infection. Now, we are not the natural host. The mouse are the natural host. The mice are natural hosts for this virus. They're fine with it. They excrete it. We pick it up accidentally. And in this case, it was because the mouse population went up. But it's a rare disease and it does not spread from person to person. It's a zoonotic spillover. Every human infection is a zoonotic spillover. And looking back at human sera, it was clear that there were cases as far back as 1959. We just didn't know what they were. Here's a summary of U.S. cases as of January 2017, 728 cases. And you can see most states have cases, the numbers in parens. We had a couple here in New York. And some of these happen when people go camping and they put their tent on these wooden platforms and the mice are underneath it and they inhale aerosolized feces. Many cases in homes where mice have dropped feces. So if you have mice in your house or apartment and you have feces, don't just sweep it up because you're going to aerosolize the dried feces. You want to wet it first so that you don't put the virus in the air. You should be careful. Anyway, this virus is not just in the deer mouse, it's in the white-footed mouse, it's in the rice rat and the cotton rat. And here's the range of Paramyscus maniculatus in the US and Canada and Mexico. So very large range, one of the many hosts of these hantaviruses. So will this become a human virus? Who knows? Who knows if the changes will be sustained? If some mouse somewhere will have a virus that can infect people, there probably already are. It's just a matter of encounters. It has to happen. And the more our population grows, the more we're going to encounter mice. In fact, we're going to see shortly that bats are a big source of viruses. So are mice. Bats are the second most numerous mammal on the planet after humans. But mice are numerous also. And we don't have really an idea of the viruses that they carry and what kind of threat they may pose to us. Nobody wants to spend money to learn this. Okay, now let's talk about bats and viruses. Bats are a wonderful is not the right word, a major source of zoonotic infections. This is a flying fox. Look at this. This can be 3 feet from top to bottom. It's Dracula embodied. Many, many new viruses have been found in bats in particular. Flying foxes like this one have had 2 major human pathogens, Nipah and Hendra viruses found in them. These cause disease not only in humans, but in horses and pigs. And a study done in 2014 found 66 new paramyxoviruses from 119 bat and rodent species. And that's just the tip of the iceberg. So Nipah and Hendra are paramyxoviruses. Do you remember a paramyxovirus? Measles virus, envelope negative strand RNA virus. Let's explore this a bit. Let's start with Hendra virus. Hendra is a town on the northeast coast of Australia. In 1994, some racehorses started getting sick. They got encephalitis, and 14 of them died. And one of their trainers, racehorses you take care of, you get close to them, you can get infected with their viruses. And one trainer died. And this virus was named Hendra virus after the town. I guess they don't mind having a virus named after them. It turned out that Hendra virus was present in those bats we just saw, flying foxes. And what we did was we built horse stables near where the bats live. And the bats fly into the stables and contaminate them with their viruses. The horses get infected, and then the horses spread the viruses to humans. And these infections continue, and they continued for a number of years. Finally, a vaccine was developed for horses specifically because there are not enough cases in humans to justify making a human vaccine. But, racehorses are quite expensive and worth a lot of money, and people want to protect them. So a vaccine was developed to protect the horses, which also protects people, right? And that's called a one world health approach. One world means we're all in this together, humans and non-human animals. And if we protect one, we protect the other. And so this Hendra vaccine is a one world vaccine. By the way, this map shows the outbreaks of Hendra virus. They've been along the coast of Australia here, not just in Hendra. And the virus we're going to talk about next, Nipah, has also been in this area and bounded by the blue line. This is the range of the flying foxes. Flying foxes live in all this area within here. So potentially all these countries and areas are at risk for infection with Hendra and the next virus we'll talk about, which is Nipah virus. So Nipah virus caused an outbreak in Malaysia and Singapore in 1998 in pig farms. So it started in the Perak region of Malaysia up here. Suddenly pigs were getting sick, displaying weird respiratory and neurological disease. It spread south in Malaysia here. And then eventually there was an outbreak in Singapore. And what happened was Singapore was importing pigs from Malaysia. They were importing infected pigs. They didn't know about it. And eventually this was stopped by killing 1 million pigs, essentially, in Malaysia, which was highly unfortunate because it's a big source of protein for the country. But this stopped the outbreak, and there are 105 human deaths associated with that, people taking care of the pigs and their families and so forth. I went to a meeting in December in Singapore, which was the 20th year after the discovery of Nipah virus, and all the science that was being done on the virus since then. Quite interesting. And again, what happens here is this virus is present in fruit bats, the flying foxes that we encountered. They're fine, just like they're fine with Hendra virus. They excrete the virus in their urine. These pig pens are near where the bats live. And in fact, next lecture, we'll talk about the ecology and what went on here to make the bats fly near the pig pens. The farmers had mango trees near the pig pens, and the bats flew to the mango trees to eat. They're fruit bats. They like to eat fruit. So the bats spread the virus to pigs. The pigs spread them to humans, and the humans got sick. So that has been stopped. So there's no more Nipah in pigs. They've taken measures to make sure the bats don't get into the piggeries. However, these infections continue in India and Bangladesh, and it's been traced to the consumption of date palm sap. So palm trees have sap, of course, like trees do, and people like to drink it. So they put these taps in the trees and they leave them on for a couple of days. And it turns out the bats like to come at night and drink the sap as well. And while they're drinking, they're urinating into the sap and putting Nipah virus in, and that gives you outbreaks of Nipah in these countries. There are some outbreaks, however, that are not connected with date palm sap consumption. So there's some other kind of contact between bats and people that's going on here that we don't know about. And in some cases, there are examples of human to human transmission, but not extensive change. Eventually, they die. So these don't become human infections. They don't become human viruses, but maybe they might at one point when they sustain the right changes. Who knows? So we should be ready. But most of the world doesn't worry about this because the fruit bats are not in many other places. But it's clearly a threat to India and Bangladesh, and it's difficult for them to develop their own vaccines and antivirals. And so a nonprofit called CEPI, CEPI.org, a number of years ago raised money and has developed a vaccine for Nipah virus, which they have funded on donations. And they're going to bring it through a phase 1 trial and be ready for an outbreak. And this is what we need for the future. We can't depend on for-profit companies to make drugs and vaccines to save our lives because they only care if they can make money. And this nonprofit approach, this is going to gain traction now with the current outbreak because we should have been ready for this as well. So let's continue with bats. Ebola viruses. Ebola virus is a member of the family Filoviridae. And the first outbreaks with virus of this family occurred in Germany, actually. They had imported primates for research from Uganda. And the primates were infected with Marburg virus. That's where the primate lab was in Germany, Marburg. So they called the virus Marburg virus, and it killed a number of workers. And that was our first introduction to the filoviruses. And subsequently, we learned that the monkeys imported had caused that outbreak. And then in 1976, an outbreak by another filovirus, Ebola virus, in DRC in Sudan. And I interviewed Fred Murphy, who was involved with the CDC at taking the first pictures of Ebola virus. You may have seen these iconic filamentous pictures, electron micrographs. He took those pictures. He's now a retired virologist in Maryland. And one day when people lose interest in coronas, I'll release that TWIV. It's a fascinating story. Anyway, there have been outbreaks of Ebola virus over the years, mostly in Africa, mostly in Central Africa, rarely elsewhere. The one in Eastern Africa was very unusual, as you will see. But these have been relatively small, a few hundred cases. And then the one in 2015 in East Africa was over 26,000 cases. And there are 4 species of Ebola viruses. Most of the outbreaks are the Zaire Ebola virus here in red, and Sudan, a few here. DRC, there's currently an ongoing outbreak. And every outbreak is started by a new zoonotic spillover. Again, this does not become a human virus. These are dead-end infections. So Ebola viruses, of course, are filoviridae. They're filamentous, very unusual for eukaryotic virus. Negative strand RNA genome enveloped particles with a glycoprotein, and the glycoprotein is against which you would like to make your antibodies. So the vaccine we talked about, which is licensed, Ervebo, is the glycoprotein put in a VSV vector. Now, how do humans get infected with Ebola virus? This is a classic zoonosis. You have an index case, typically a contact with an animal carcass, not always identified, but we think that's the case. Bushmeat. People in Africa don't have always nice shopping centers to purchase their food at, so they hunt their food and they will eat primates, they will non-human primates, they will eat bats, rodents, and if these have Ebola viruses in them, they start an outbreak. So the index case has contact, and then the virus is transmitted to others by close contact with infected fluids, typically families and doctors and nurses that care for the sick people. These chains of infection are typically short, R naught of about 2. They require close contact. No droplet transmission. Few here. DRC, there's currently an ongoing outbreak. And every outbreak is started by a new zoonotic spillover. Again, this does not become a human virus. These are dead-end infections. So Ebola viruses, of course, are filoviridae. They're filamentous, very unusual for eukaryotic virus. Negative strand RNA genome enveloped particles with a glycoprotein, and glycoprotein is against which you would like to make your antibodies. So the vaccine we talked about, which is licensed, Ervebo, is the glycoprotein put in a VSV vector. Now, how do humans get infected with Ebola virus? This is a classic zoonosis. You have an index case, typically a contact with an animal carcass, not always identified, but we think that's the case. Bushmeat. People in Africa don't have always nice shopping centers to purchase their food at, so they hunt their food and they will eat primates, they will non-human primates, they will eat bats, rodents, and if these have Ebola viruses in them, they start an outbreak. So the index case has contact, and then the virus is transmitted to others by close contact with infected fluids, typically families and doctors and nurses that care for the sick people. These chains of infection are typically short, R naught of about 2. They require close contact. No droplet transmission like SARS-CoV-2. Although if you're intubating someone and you're making an aerosol, that aerosol could spray some distance and hit a close by healthcare worker, for example. Some examples of how these start in 1996 in Gabon, 37 cases. A chimp found dead in the forest was eaten by people hunting for food. And then 18 people who were involved in butchering the animal became ill, and 10 other cases occurred in their family members. This is a very typical scenario. Family members get infected. And if the outbreak can be identified, it can be contained. You do infection control, and you can limit the outbreak. But these are all very close contact with one another. Gabon 96.7, again, 60 cases. The index was a hunter who lived in a forest camp. A dead chimp was found in the forest at the time and it found to be infected with Ebola virus. But again, we don't know if that particular case, the hunter actually had eaten or touched that dead chimp. We're unable to get that information. So there's always this circumstantial evidence, but we don't often have a smoking gun to identify where the virus came from. Now, in 2015, there was a huge, the biggest ever outbreak in West Africa, Guinea, Sierra Leone, Liberia, but it began here in Guinea in December 2013 to February 2014. And so the first case was this 2-year-old child who got sick on December 2nd and died 4 days later. Fever, black stool vomiting. And that child transmitted it to her sister, her mother, her grandmother, a nurse, and a village midwife. They all died. The midwife transmitted it to other villages, there to the left, there down here, and that started other chains of infection. So all of this wonderful epidemiology, this contact tracing was done to sort this out after the fact. In fact, it was 3 months before anyone knew this was Ebola virus. And by then, this had spread extensively because it had never happened in this part of Africa before. So it wasn't thought to be an issue. So they didn't know what this was. They thought it could be malaria or some other hemorrhagic disease. 3 months later, finally, they figure out it's Ebola. And by then, so many people are infected that you get all these transmissions and short chains of infection. But you can see family members, healthcare workers, and funerals. People attending funerals, often these people prepare the bodies of their family members. They get infected. And that's how you control infection. You control it in the hospital, you control it at home, you control it in the burial stage. So this ended up being over 26,000 cases. It was a real serious outbreak. Now, this virus, we think, comes from bats, but very difficult to prove. The Marburg virus, which was involved in that German outbreak in a primate center, the virus itself has been isolated from the cave-dwelling fruit bat, Rosetus aegyptiacus. So there the virus has been isolated. So that's pretty clear that that bat is a host for Marburg. And so this bat must transmit it to primates and infect them. But the problem is Ebola viruses. We're not sure. The RNA and antibodies to the virus have been found in 3 tree roosting bats. They haven't found infectious virus. It's very difficult to do these studies. It's not easy to catch bats. It's dangerous. You have to go into caves and you could get infected, so you have to be all suited up. So I'm not sure we'll ever have infectious virus, but the idea is that it's likely in a bat or bats, and humans, gorillas, chimps are dead-end hosts. We know that chimps are killed by it, gorillas are killed by it, and somehow it goes from one to the other. And this is the scenario. Bats are the reservoir, multiple reservoirs, possibly different species, and these can spill over periodically into chimps and gorillas, who then can infect humans. So we can get it by bushmeat hunting of chimp and gorilla, or even bat hunting for bats. And if they're infected, we get the infection. And then we transmit it in short chains among each of us. And so that's the current idea for that. But the key here is that every infection is a brand new spillover. As I said, human to human transmission, contact with infected blood or body fluids. All of these fluids have virus in them, typically from a very sick person or someone who has died during burial preparations. Also, needles, syringes have transmitted the disease, and it's not by insects, not by water, not by food, not by aerosol. So none of these droplets will transmit unless, as I said, you're doing a medical procedure and you generate these large droplets. If you're standing right over a patient and they expel these droplets onto you, you will be infected, but it's not what we consider aerosol transmission. It's a procedure. If you're walking around breathing, you're not going to transmit Ebola virus. The virus enters at mucosal surfaces, respiratory, for example, or if you have breaks in your skin, it can be injected by a needle, and you can find, again, virus in all of these fluids. And so contact with any of those will transmit infection. Incubation period ranging from 2 to 21 days. And during this period, you're not contagious. You're not shedding virus in sufficient quantities to infect someone else. Very different from SARS-CoV-2, 1 to 14-day incubation, and in many cases, you're shedding virus. Multi-system infection, systemic involvement, gastrointestinal involvement, respiratory system, vascular system, neurological system. The virus not only replicates in many, many tissues, but also induces an immune response that is overabundant and can't be controlled. 30 to 90% case fatality ratio in Africa. We know that you can actually have a lower case fatality ratio by proper medical care. So if these patients are cared for in a certain way, they can be saved. But this is a very typical feature of a zoonotic spillover, a virus going into a brand new host where it's not evolved for many years, and very, very serious disease. Okay, another bat infection, SARS-1, classic SARS. This begins in February 2003. This is a text of a ProMed Mail post. If you haven't looked at promedmail.org, wonderful place to learn all about outbreaks of everything that are happening in the world. In this February, a doctor had sent an email. This morning, I received an email and searched your archives on ProMed, found nothing that pertained to it. Does anyone know anything about this problem? And here was the email this doc had received. Have you heard of an epidemic in Guangzhou? An acquaintance of mine from a teacher's chat room lives there and reports that the hospitals have been closed and people are dying. February, nobody knew anything about this. That's the first information we got about SARS. So SARS is severe acute respiratory syndrome, which now nobody needs to be told, but back then no one knew about it and we forgot quickly. It began with an outbreak of severe atypical pneumonia, unknown etiology in the south of China in November. So we didn't find out until months later. 305 cases, 5 deaths initially. Short incubation period, fever, chills, headache, malaise, myalgia, typical flu-like syndromes, and then moving into another phase, which included shortness of breath, and 10 to 20% of the patients may require mechanical ventilation. Sound familiar? So that's the beginning, November 2002. Now, in February, this Chinese doctor had treated some of the first patients in the outbreak. He traveled to Hong Kong on February 21st, 2003, stayed on the 9th floor of the Metropole Hotel. This is the Metropole Hotel. It has become infamous because of this incident. The next day, he died in the hospital. He got sick quickly and went to the hospital. He died. However, in the short time he was there, he spread his infection to 10 other people in the hotel, and they flew on to Singapore, Vietnam, Canada, the US before they had symptoms, and they brought the virus to those different countries. And he probably just walked by them or was in the elevator with them. He was a super spreader. He infected 10 people. That's the definition of a super spreader. He was probably shedding a lot of virus and infected them readily. Eventually, this infection spread to 8,000 people, 29 countries, 10% mortality. I like to show this travel poster, which was issued at the time. Hong Kong will take your breath away. Indeed. So what is the outbreak all about? Here's the case reports. This is the epi curve. The number of cases you can see from February 23 peaking in March. Outbreak is over July 2003. Never seen again. The virus never found again anywhere except in labs in the world. And that's why people, I guess, the beginning of SARS-CoV-2 didn't take it all that seriously, because this one had ended. We thought that this one would end as well. So here are the cases, 8,000, 10% case fatality. Vast majority, of course, in China, Hong Kong, a few other Asian countries, Australia, just a few, Europe, very few, US, very few. So this is really very different, of course, from what's happening now. Now, this virus was eventually isolated. It turned out to be a coronavirus, which was a shock to everyone at the time. Why? Because we'd known of 2 coronaviruses up until this point. They were isolated in the 60s. They caused common colds. This is the first time an epidemic coronavirus that caused serious illness had been identified. So this awakened the world to coronaviruses. Where did this virus come from? Well, human sera that were collected before the outbreak, this was all done after it, of course, they were found not to contain antibodies to SARS coronavirus. So this was something new. And the earliest cases in the south of China were in handlers of animals for the open food market, where they sell meat, typically bringing meat in from farms or from animals that are captured, and they're butchered and they are sold. And these people are obviously exposed to blood and they can get infected. So they had the earliest cases. And these animal traders had significantly higher prevalence of antibodies to the virus than control group. So this said, it must be in one of these animals in these meat markets. And so the story evolved even further. So people started doing wildlife studies, and they found in caves in Yunnan province, highly diverse SARS-related coronaviruses in horseshoe bats. This is a horseshoe bat right here. All the genetic diversity needed to make SARS coronavirus were found in this one cave. So you have the genome of SARS-CoV, and you can find all its pieces in viruses isolated from bats in this cave. However, no one found any one SARS coronavirus in any bat that was exactly the same or close to what was in people. That, coupled with the fact there were no SARS cases in Yunnan province, okay, that's where the cave is with all this viral diversity, no SARS cases during the outbreak. So obviously it started in the south. So the hypothesis emerged that the progenitor emerged in bats by mutation and recombination. And then the bats infected civet cats. So the civet cats are sold in the market. These were found to be positive for a virus virtually identical to that which infects humans. So the civet cats were thought to be infected in the countryside by the bats. They were brought to the market, and then people, the handlers, the meat handlers were infected by the civet cats, and that started the outbreak. And that's because, again, this virus is not found in the countryside. So it was brought in, and so the scenario is bat, civet cat, SARS-CoV. So that's a horseshoe bat, and that's a civet cat. All right, let me see before we go on if there are a lot of questions here. How do programs like PREDICT effectively sample for these quasi-species? They're not perfect. So they look at wildlife, and they're limited in what they can do. Sequencing that doesn't reveal the quasi-species. They can just say, what's an average sequence? And then you can ask, do these viruses have the potential to infect human cells? So documented transmission of a plant virus to humans. I don't know of any, no. Are viruses with higher mutation rates more likely to establish in new species? Yes, most of these zoonotic spillovers are RNA viruses. Why can't someone with hantavirus spread virus to other people? You don't make enough virus. The virus titers aren't high enough in respiratory secretions. It has to be well adapted to replicate in you. It has to replicate the high titers, and these viruses haven't done that. If human is the dead end, why did doctors need to track down every infectious case in quarantine? Because there is close contact in families, healthcare workers caring for patients, and they didn't know how to do infection control. So we had to track the cases and say, we have to isolate these patients. So I know it's misleading to say it's a dead end, but if you contain those cases in hospital and you teach people not to touch the corpses when they bury and so forth, you can stop the infection because it's not able to transmit effectively. You have to have very close contact for it to transmit. How long can a virus particle remain infectious in droppings? Well, it depends on the virus. If it's enveloped, maybe a few days. If it's not enveloped, maybe months. How long can they survive on dead animal? Same thing. Weeks to months to years, depending on the virus. Would you agree with the suggestion of Peter Daszak that humans should stop eating bats? I don't think you can say that eating bats caused the outbreak. For the current virus, it might have happened in another way, and we'll talk about that. These viruses don't necessarily come over by eating bats. If you stopped eating bats, I think there would still be spillovers. There are other ways. Do so many human viruses trace back to bats just because of the large size of the bat population? Yes, bats are 20% of all mammals, the most numerous mammals. They fly, so they have a big range, and they have more viruses than any other thing on the planet. Have people proposed a naming system for viruses separated from the location where they're identified? Yes, and then most viruses are named that way now. Is super spreading a function of merely social habits, or is there something biological to it? Yeah, it's shedding. Super spreaders appear to make more virus per cell than other people, and therefore your droplets have more virus in them. And even the tiniest droplets that you make that go farther seem to have more virus than normal, and that's why they're super spreaders. How can you get viruses from bushmeat if you cook it? Before you cook it, you have to prepare it, right? And that's when you contaminate yourself. It's like chicken. Chicken is full of bacteria. if you don't wash your hands after pulling the chicken out of the package, you're going to get infected. Same thing. All right, the next coronavirus, MERS coronavirus. SARS-1 is gone. 2012, 60-year-old male patient dies of pneumonia and kidney failure. They get a virus out of him. They sequence the genome. It's another coronavirus. It's not SARS, but it's a coronavirus. It binds a different receptor. So the SARS coronavirus receptor is ACE2, angiotensin converting enzyme 2. And this one is dipeptidylpeptidase 4, another membrane-bound enzyme. Very interesting. So they call this Middle East Respiratory Syndrome coronavirus. And again, phylogeny, by now we have a bunch of coronaviruses from bats. It's closely related to bat coronaviruses. And so this case turns out that the virus went from bats to camels at some point. And then in camels, it is endemic, and particularly dromedary camels, there with the one hump. All the camels in the Middle East and Africa appear to have MERS-like coronaviruses. Camels get infected at a young age when they're born. They get a mild respiratory disease, and then they recover and they're okay. So they got this from bats a long time ago. You can trace the phylogeny. Then the camels spread it to humans. So most of these infections have been in the Middle East, of humans anyway. And there they raise camels, they race them, they have them as pets, they eat them. So there's a lot of human-camel contact, and so that's how the infection is generally spread. Typically, most of the patients have other healthcare issues, but not all infections have a camel source. So we think there's some people infected without symptoms that are spreading it, but we've never been able to identify that. We don't know how it's transmitted to them if it's not a camel source. We don't know why so few humans are infected. It's not adapted to humans yet. It hasn't become a human virus. Every new case of this virus is a new spillover from camels. So it does not go human to human. So lots of interesting questions here. Whether it will, it's been going since 2000. What was the date? 2012. You'll see how many cases we have so far. Will it adapt to humans? Who knows? It might. I think we should be ready. We have a camel.
My bad.
Vaccine, which is being tested. Is there not enough cases?
I knew it. Well, I was about to jump the gun, but I knew he was going to say it. So, I'm going to bump it back just a little bit.
So lots of interesting questions here. Whether it will, it's been going since 2000. What was the date? 2012. You'll see how many cases we have so far. Will it adapt to humans? Who knows? It might. I think we should be ready. We have a camel vaccine, which is being tested. Because there are not enough cases of human disease to justify a human vaccine, but every camel is infected, so you could vaccinate them and prevent spread to human. Another One Health approach, right? That would work. So this is still in testing. Whether it's going to be widely accepted is not clear. Because again, the camels don't really get sick, so the people who are raising them may be reluctant to use it if they have to pay for it and it doesn't help them in any way. So that should be interesting. So here's the transmission and geographic range. As the most recent numbers I could get through 2019, 2,500 cases, 858 deaths in 27 countries. And that's a 34% case fatality ratio. And you can see the epi curves, they're bumps of cases. And it's been going down as we've getting better at recognizing and containing the infection. And these colors tell you how these infections occur. Most of them are camel. Well, camel to camel is the orange. I mean, camels in all these orange regions are positive for MERS coronavirus. So a lot of camels. And then camel to human is the red, mainly in the Arabian Peninsula, and short human to human chains, right? But then some infections have been exported to other countries, Morocco, parts of Europe, US. There was one in Korea, I believe it was back here. That accounts for this big spike. Oh, sorry, Korea is the red one here. See, so one patient was infected in Saudi Arabia, flew to Korea. He was fine. He got sick. He went into a hospital, and they didn't know what was wrong with him. So he infected lots of other people in hospital, and they went home and infected people. So that was that. until they figured out how to contain it. So he was probably a super spreader. So this human is typically in healthcare settings where they don't recognize the infection soon enough to contain it. Whether or not this becomes a human virus, we don't know. But I think we should have antivirals in case, but we really don't have anything that's been shown to be effective. And that brings us to how we started this course. SARS-CoV-2, the first pandemic of the 21st century, still ongoing as we speak here in April. Started with a cluster of pneumonia cases of unknown cause in Wuhan, December 2019. And initially, the Huanan seafood market was thought to be the beginning. Now, why? Because, well, SARS-1 started in an open meat market. Here at the seafood market, they sell meats as well as seafood. So they said, okay, maybe it happened here. And the first 40 cases or so are mapped on this epi curve, number of cases, and whether or not there was Huanan seafood market exposure. The very first case, December 1st, no seafood market exposure. And you can see almost half of the other cases, no seafood market exposure. But all the ones that were connected led people to think that it was the beginning, but it really was not the beginning. It's now accepted that this was not the beginning. Probably someone went there and spread the infection, but it started somewhere else. And as of today, 2.5 million reported cases globally, 165,000 deaths. So this virus, of course, spread extensively within China despite a lockdown and out of the country as well. And it has now reached many, many countries, and we're all trying to deal with this in different ways. The R0 is between 2 and 3. It's effectively spread. And because 80% of infections are mild, this spreads silently. And you don't know that you're spreading it. You don't feel sick or you feel like you have a cold or just flu, and you're spreading the virus. So very difficult, very different from SARS-1, where most patients were hospitalized. Over 50% of the infections ended up in hospital, and we could contain them there. And that's why that one was burned out. But this one has an insidious way of spreading because most of the infections are quite mild. Where did this one come from? From the earliest cases in Wuhan, the virus was isolated, the genome was sequenced, and here it is on a phylogenetic tree of other alpha and beta coronaviruses. And I just want to point out, coronaviruses of many different animals, and you can see here human coronavirus OC43, 229E. These are common cold coronaviruses. NL63, there are 4 of them now. HKU1, that's the 4th. They're quite distant from the original SARS-CoV-2. And here's SARS coronavirus isolate and a CIVIT. The CIVIT isolate, you can see how close they are. SARS-1 and CIVITs are.
All right. I'm letting y'all know that this is his work. What you see on the screen, this right here, is where he mapped this thing out himself. Submitted his work to the University of Columbia and other, other peer-reviewed journals, and it's there. So y'all remember 2 videos ago I talked about, well, you heard him say, but you heard him say in a lecture, that he mapped out the genome, right? This is the work that he does.
SARS and some bat SARS-like viruses. But here the SARS-CoV-2 is in a different branch of this tree. Not really that close to SARS-1, 80-some% genome identity, but this virus has 96% nucleotide identity with a bat coronavirus called RATG13. This was identified, look at it right there, right next there on the same branch. This virus was identified in 2013 in a bat cave in Yunnan province, 1,000 miles outside of Wuhan. And so this SARS-CoV-2 and RATG13 have a common ancestor. SARS-CoV-2 was not derived from RATG13. It couldn't have been. It's 4% difference, and this virus, RATG13, was identified in 2013. It's too long ago. Something else is the immediate precursor. We don't have it, but it clearly shares an ancestor with RATG13. This is a bat virus. There's no doubt about it. It wasn't made in a lab. It wasn't escaped from a lab. It came from nature. And a recent interesting analysis of recombination, it's a preprint found here. This is by Columbia professor Raul Rabadán from his laboratory. shows that a recombination was clearly involved in an ancestor of this virus to generate it. And this recombination encompasses the receptor binding domain. So here's the spike glycoprotein of SARS-CoV-2. And remember, there's a domain called the receptor binding domain here in green. This binds ACE2 receptor. And this virus has high affinity RBD for ACE2. RATG13, which is the closest, has an RBD that can bind ACE2, but it's much lower affinity. So mutations, amino acid changes have been selected in SARS-CoV-2 that allow high affinity binding. And this RBD, by the way, can be tracked by recombination. So if you look at the phylogeny of the whole genome, you find that this SARS-CoV-2, the closest relative is bat RATG13. And human SARS is a bit farther away. But if you just look at the RBD in terms of phylogeny, now RATG13, SARS-CoV-2, and human SARS all map together very closely on this phylogeny. And so Raul has come up with this scenario where there were recombination events in ancestors. And so here in red, there was possibly one before 2009 that introduced the receptor binding domain into RATG13 and the precursor of SARS-CoV-2. And so we can see that today in RATG13, and it further evolved in SARS-CoV-2 to bind ACE2 very highly affinity, with a very high affinity. And where that happened, we don't know. It had to happen in an animal with a human ACE2-like receptor. And the other possibility is that the recombination event actually occurred before SARS-1 in 1992 or before. And this kind of analysis you can do by looking at recombination events and phylogenies and plotting them with time. So it's quite clear that this is a virus of bad origin. How it got into Wuhan, we don't know. But sampling of wildlife for these viruses will likely provide an answer. And we need to do more of that. And so here's a summary of the origin of these human coronaviruses. They all came from nature. None of them came from a lab. You hear lots of conspiracy theories in the press. It came from a lab. Someone made it or someone worked with it and it got out. It's all baloney. It's all wrong. There are people who are doing that who don't understand virus evolution and phylogenetic analysis. And so the 4 human common cold coronaviruses, 63229, OC43, HKU1, they're all spillovers from animal viruses. 2 from bats, and 1 went through an intermediate host. The other is from a rodent virus to a cow to a human. And the other, we don't know what the intermediate is. But you see the pattern here. We always think the origin is one animal, and then it went through something else before getting to human. And question marks, we don't know what those intermediates are. SARS-CoV-1, bat to civet to human. MERS-CoV, bat, camel, human. There's even a pig coronavirus, severe acute diarrhea syndrome coronavirus, emerged in China a couple of years ago. It's a bat virus that became adapted to pigs and causes diarrhea in pigs. And then we have SARS-CoV-2, clearly a bat virus, now a human virus. If there is an intermediate host, we don't know it. Now, someone said, why don't we stop eating bats? It's not clear that eating bats caused this. You see, none of these others was a direct introduction of virus into humans. We have to find this intermediate, and that would solve this problem. But even if it turned out to go from bats to people, you don't have to eat the bat to get the infection. Many farmers in the country in China harvest bat guano from caves to use as fertilizer in their fields. And it could be that one of them got infected and brought the virus into Wuhan. We just don't know. So, making blanket statements about cultural practices, I don't think is useful. I think we need to sample wildlife and figure out this problem. A few last words here before I close. First of all, I went to Australia in 2014 and I interviewed Linfa Wang, who has since moved to Singapore, and I was his guest back in December. He studies bats and the viruses that infect them. Fascinating guy. Old Twiv, he's the real Batman. It's really cool. He's so smart and really neat to hear his ideas about bats and viruses. And he's currently very tied up, of course, SARS-CoV-2 in Singapore. And the other thing is, a lot of these viruses, how do you work on them? The highest containment laboratory is a BSL-4. And here you have to work on viruses like Nipah and Hendra and Ebola. Not SARS-CoV-2, you can work in BSL-3. But BSL-4, you have to wear these spacesuits. You have to have oxygen or air pumped into them. You have to go through an entry and exit procedure. And I was fortunate with some of my TWIV co-hosts to do a documentary in a BSL-4 in Boston called The Needle, National Emerging Infectious Diseases Laboratory. And so we called it, of course, Threading the Needle. You should really watch it. It is so cool. Nobody has ever done this because this lab was not open yet. So we were able to go in and film this documentary. Very cool. And so viruses with high mortality, person-to-person transmission, no approved vaccine or antiviral, those are the ones you have to work on. And we have a number of these in the US, and people are working on viruses like Ebola and Lassa and Nipah and Hendra in them. One last series of thoughts for you. So we've talked about, how these emerging infections occur, what they do to people. How common are they? Well, I think dead ends are very common, as I said before. I think we're all infected frequently with viruses that never go anywhere. So ones that produce sustaining transmissions are very, very rare. So what we see today is an incredibly rare event, which just happened to have produced a virus in nature that encountered a human that spread. We can't predict them. We cannot predict what's going to happen, but we can know what's out there. We can sample wildlife and know what's out there, what has pandemic potential. And we can react. We can be prepared. We can say, let's have a pandemic plan. And if a new virus emerges, we're going to make sure we have personal protective equipment. We're going to make sure we have hospital capacity. We're going to make sure we have diagnostic capacity of all sorts. We had none of those. We have none of those for this outbreak. We were completely unprepared. And it's a shame because the US and many other countries of the world are technologically wonderful. We can do any of this, and we didn't because we didn't want to spend the money. And it's a shame that people have to die because of that. It is really the tragedy here that all of this could have been prevented. It's not anyone's fault. It just could have been prevented very soon after the first emergence in China. For example, we could have had pan-coronavirus antivirals. We talked about this briefly a couple of lectures ago. It would have been easy to sample all those SARS-like coronaviruses in bats and look at their RNA polymerases. They're the most conserved protein in all these viruses. It would be a piece of cake to make a pan-RDRP antiviral. Nobody wanted to do it. Why not? Companies weren't interested. There was no money to make. Now everybody's interested in antivirals and vaccines because there's money to make. It's kind of sad, isn't it, that our health depends on for-profit companies and they won't do anything for us until they can make money. So I think we have to change the way we make drugs and vaccines. We have to have nonprofits like CEPI doing it. And this week I'm going to talk to a number of virologists who are starting another one of those nonprofits to try and be ready for the next pandemic. All right, so we're actually going to continue some of these ideas next time. There's a brand new lecture for this course, Ecology of Viruses, because spillovers depend a lot on the interaction of animals and viruses with their...
All right, so there we have it. Some daggers in there. I did not want to make this 3 hours long, but it ended up being that because Dave sent us on a wild goose chase. By the way, I continued to keep on digging while the lecture was going, right? And I can't find this lady, yo. I cannot find this lady. I can't. I'm sorry. I don't know who this lady is claiming to be a nurse. She is unfindable. Can't find her. Nope. But hold on, let me blow my screen up. Hey man, what y'all talking about in there, man? What's going on in the world? Y'all can unmute, man. Satep, I see you in the building, bro. What's good? What y'all think about that lecture? From what y'all heard or could hear?
Yeah, that's right. That's right on point right there. You know what I'm saying? Like, and you can't act like you really know that information, because who finna sit there and deal with some viruses for an hour and a half? What'd you say?
I was making sure my mic still worked.
Oh, but yeah, man, that's it. That's all I had. Like, we got to sit down and learn, ask questions. That's what I was saying in the chat. Like, the only way you learn is ask questions. And the more you're skeptical about something, it's better to go ahead and investigate it thoroughly. Get all the information. Don't just get the information, the confirmation bias, where somebody's saying what you think it is. Get all this information so that you can understand what it is that you're dealing with. And this is just a video of him showing and talking to you about it. Like, imagine if you was in the field like him, just kind of going in and really understanding this thing through and through. You know what I'm saying? So we got to pay our respects to the professionals that are doing that. There's too many diseases out here, man.
Mm-hmm. Way too many. Ones we ain't even uncovered. Yo, so before I go again, man, I do want to put this up here. I am going to look in the chat for questions, though. I ain't just going to dip on y'all, but you know what I'm saying? I just deleted one of them off my file. But for anybody interested, we showed y'all how to go look up every nurse in your state. It ain't necessarily got to be Tennessee. You can go. Now, let's do ourselves a favor. Let's go see if we can find Malcolm on here. Let's see if we can go find Malcolm on here. Now, I know he listening in. You know what I mean? He said he was on here. Oh, shoot. I done messed it up. But we're going to see if we can find Malcolm on here. So, Tepper, what you're looking at is a list of nurses who practicing nurses, no matter what field, whether it's an RN, whether it's not practicing, whether it's an LPN, you know what I'm saying?
You might see me on there.
I didn't look. I think I'm getting this thing again, man, because I didn't straight up. I didn't deleted the other file, which probably was attached to this file. Now I'm gonna be searching. But anyway, hold on one second. Kick me out, Satel.
With pleasure. Kick Shawn out, y'all. Man, back.
No, I'll be right here.
Oh, okay. His computer must.
What up, Satel? What you on, man?
Nothing, I'm just clearing my throat. I was laying down. Yeah, I was laying down. Just getting a little rest before we get the we got the coffee and cookie tonight. Talking about the concentrates over there on Coffee and Cookie Podcast. Y'all go join that. Like, subscribe. Talking scientific. What's up with you, T'Challa?
Man, hooked up, started me another batch. Speaking of which, of tincture, doing room temperature. I'm doing a couple of different ways to see what my tolerance is and things like that.
Okay. Yeah.
Just because the cold thing, you want to leave that in there.
Right. You need something now.
So.
Right. Like, shit, I want to see the fruits of this labor now.
And just getting a training session in with this 3-hour video.
Yeah, that's what's up, man. What's up?
We definitely appreciate y'all rocking with us, though, man. These bills be long, but these kind of bills, they be needing to be long.
Yeah, definitely. Can't just glaze over this like a Krispy Kreme. Real.
Mm-hmm.
Oh, this down.
I think Sue in the building.
Where he at? Oh, okay. He said to somebody. Yeah, you say cannabis 3 times, man. You gonna see Sutek appear. He gonna come out of nowhere, y'all. It's down there like if you say evolution too many times around. He just appeared, man.
Man, that mother was trying to download again, man. That file so thick. What's crazy?
About to mess your whole Mac up, huh?
Nah, bro, I'm deleting this joke. You see, I'm doing it right now. I ain't even finna get into trying to dealing with I don't want no part of it. Trash empty. I don't want no more part of it. But anyway, yeah, so we don't find the lady name on that list, man. We did that already. We finally got that ironed out. And need to get this off my list. And so that's that on that. We ain't gotta deal with none of the foolishness again, man. Look, people want to deal with it. You know, hey, y'all can send them to this video. They can deal with it like that. We can go from there. I see Sutek in the building with coffee and cookies. He's on the coffee and cookies. That's later on today. Go subscribe to Coffee and Cookies. Nope. Yeah, look at the number one troll. Number one troll went looking, y'all. She ain't found no Facebook page either.
For what?
This lady claimed to be a RN, yo. Said she got Bell's palsy from the vaccine. Said she from Nashville and she a nurse. Now, we didn't find. We don't know what vaccine she took. We didn't find her in the nursing list in the state of Tennessee. She ain't got no Facebook page because Mika been looking for it. But all we got is a YouTube video that just suddenly popped up as being shared, but it all comes from some Pegasus account where it originated. So, you know what I'm saying? That's where we at from that. But the number one troll even went looking. You know that troll was gonna look.
Oh.
She about the drama. So that's pretty much, man. That's basically we got hit with the curveball. Dave sent us on the goose chase for about an hour and a half. That's why we at 3 hours right now. But anybody who made it to this end of the video, you know what I'm saying? Oh, and Twitter removed the video, of course, because there's no documented information for that, man. If you had something like that, you're not going to go on Twitter and make a video. You're going to go. Y'all remember?
Hey, look, Shawn. They can get paid for that. That's a story that news people will pay for. They're not just gonna give it away for free.
Right. Yeah. And you absolutely damn. I didn't even think about that. You're right. They'll pay hella.
All that. They paying for that.
You're right. They get hella money for that. Yo, and look, but y'all remember like on the earlier shows, like either episode 1 or 2, 2, I think, I gave y'all the place where you could go like Pfizer. Remember, we was going through Pfizer's reports and we were breaking down the reports and the data on that. Pfizer has a document that, remember, that I put in the description, where you can click on the website and report any adverse effects directly to the company, yo. And it's also going to be registered in the database of all the doctors and everything like that. So yeah, I ain't think nothing about it though, yo. That is an excellent point, man. They will pay. Hell yeah, they will pay. Man, she probably could couple bands for that. Maybe more than a couple bands. Might get about 10 racks.
Man, she gonna get a nice hefty piece of change for some shit like that. That like I said, that's breaking news, and the news people, they want to be the first with that. So whoever break the story first, that's who get all the publicity. That's where all the traffic goes to. So yeah, she wouldn't get that up for free. No, sir, buddy. Not in 2021.
Yeah, that's too easy, you know. But we had people eating it up, you know what I'm saying, sharing the dude video. The dude getting major hits on the video. She said she got the vaccine and then 3 days later she developed Bell palsy. So that, but Tennessee nurses received the vaccine on Thursday. So you got Friday, Saturday, Sunday. Sunday would have been 3 days. However, the video was out 2 days ago. So
Sound like a Jesus rose on the third day story to me.
yeah, it would have sounded like. I wonder who paid her. From West Africa, I can't approve your message. It was released from Citizens Free Press. Okay, that one now I see that one. It was released. Let's see what they say. Citizens Free Press. What is that? Janky website. All of these things is hard. All of these things, but why would she say Nashville though? Have y'all seen this website? Look at this shenanigans. Yo, we're gonna have to do an after show, yo. This mug getting too long. Look at this. Look at this, yo. This ain't how you do stuff. Click on it again. This ain't how you do stuff. Like, for real, you playing with people's livelihood, yo. Look at that. COVID outbreak at an In-N-Out burger. How you know that? Breaking news. Trump signs COVID. President issued. Like, this is this how they rolling then, you know what I'm saying? If this how they rolling. Pork. Oh, okay. Wrestling Bar Christmas message. Yeah, I'm getting off that, bro. I might get a whole bug. But yeah, man, I mean, that's where we at with that, man. Sheesh. But yeah, so what you talking about? Yeah, nah, shenanigans. I was trying to see what Nunu was talking about. Question: can a vaccine change or alter your? Someone sent me. Oh yeah, Keisha, we did that a few videos ago. Yeah, we did a video on that right there. You just go back in the archives and grab those videos. That's the reason for the videos. That's why instead of making Facebook posts, I just started documenting them in videos because people seem to watch a video faster than they'll read a Facebook post. I mean, it's just the truth. You know, so we just deal with it like that. Like, we got the videos. Just deal with it like that. It's it. See, there go the van video right there. We deal with the 12. Look, van again. It's twice we didn't deal with the variant. 4 days apart. Fake vaccines. See, they go to Vax News right there. That's important because these got them documents on it. These episodes right here. But as far as altering DNA and all that, yeah, you're gonna find it. You're gonna find it. See, you're gonna find that video over here. So yeah, we in there. We in there. We got enough evidence to actually combat it. Ain't no part 100. What you talking about? Do a part 2? She can go back to the earliest, the first 2 videos. It don't alter the DNA. You really ain't even got to watch the videos. You can go in the description of the videos and pull the sources out. Can't remember it's gonna be in the cell membrane video. The one with the cell membranes, because when we get into the explaining what a cell membrane is and all of that, we start talking about how the vaccine work and all of that. So we did that video. We actually did the video twice, explaining how the mRNA vaccine worked, and explaining kissing the dog in the mouth, tonguing and explaining that the mRNA vaccine, you know what I'm saying? We got into the gist of how it actually work and do what it do. So that's where your video gonna be at. I'll show you again. Yeah, and it would be nice if you did, instead of listening, you did take notes, you know, like some people. You know what I'm saying? Really about the science. Yeah, you approval. Man, this dude yelling and everything. See? You start talking about the cell membranes and all of that. And we start dealing with old pseudo Ali in this video too. But yeah, I'm gonna end this one, man. We'll do a little after show on Coffee Pie, so I get ready for coffee and cookies. They're a couple of hours away. You know what I'm saying? They gotta go get their strands right. And then they're gonna come in and do what they need to do. But yeah, I mean, remember, we also listen to the doctor. But I'll bring it back up and we'll deal with it in the after show, even though we made this one super long. All right, so if you ain't subscribed to Coffee Pie Side TV, you're gonna miss out, because that's what we're gonna continue to build, and we'll just specifically deal with that one claim. Does it alter the DNA? Peace. All right, ladies and gentlemen, the books is in. The books are in. Yep. They in. They're here. Subscribe to Coffee and Cookies in the chat. You see Coffees and Cookie? Subscribe to them in the chat. Subscribe to that page right there. But we about to end up on Coffee Pie Side TV. We'll deal with does the mRNA vaccine alter your DNA? Peace.